That's great. I'm going to make sure we hear something here. Yeah, great. I'll just test quickly if we see the slides and then. Okay, so I can hear mic 1. I'm checking mic 2 right now. And I'm testing mic 3. Mic 3. Mic podium online. Check, check. Still want to check mic 1. Mic 1A, check. Good. If you want to get started, you want to get me the thing, I can put the background slide so it's like don't.
Mic 1, check. Online, yep. Mic 2, check. Sounds good. Mic 3, online. Hey, mic podium. Check, check, mic podium.
5 minutes. Not be able to finish the slides, but you can remind me like if I was a bit running out of time because I feel like at the end of my slides is not like it's important, but I can skip it. Okay, but I just want to respect the time. So it's not start the year, right? So I gotta still let you in my ear. Check for the we were like in a group, like we did all like the biplane thing on the dynamics and see you. Yeah, you Wait, is that it's happening? Yeah. Where?
All right, final mic check. Mic 1. All right, mic 2. Nice. Your mic. Podium. Salut tout le monde. Good morning. Et finalement, mic 3. Mic 3 in the room, online. Amazing.
Is this on? Hey everybody, we're gonna get started in a few minutes. So if you could wrap up grabbing your breakfast, coffee or water. If you haven't had a chance, there's food in the back corner, just behind the bulletin boards. But we're gonna get started in 2 or 3 minutes. So if you could finish grabbing your food and make your way over to your seats, we'll get the programming started. Thank you.
1, 2, 1, 2. Hello, everyone. Hello, everyone, and welcome to Montreal. So today is a very hot day. I hope that you have AC in your hotel. Also, I see the room is pretty full, so no one fell in the construction hole in the front. That's already a very good sign. But we're really happy to have you today at the conference. Yeah, we're really happy to have you today at the MoML 2024 conference happening in Montreal. So MoML was started roughly 2 years ago in Boston, and then has transitioning like between Boston and Montreal twice a year, and it attracts like a lot of people, like more than 200 people each time, twice a year. That regroups a lot of talent that work at the intersection of molecular discovery and AI. We get a lot of posters about drug discovery, but also about material design and other applications as well. Some more theoretical, some more applied. It's really great to have a lot of diversity in the talks and the posters. The MoML conference was first introduced like in memory of Octavian, who was a postdoc at MIT and a close friend to many of us here. And we continue to keep the tradition of presenting this conference as in memory of Octavian and the great work that he did in molecular machine learning. So for today's schedule, we had some last minute changes that happened due to a cancellation. We were able to find an author last minute, like during the last 5 minutes. So no worries, the schedule remains the same, but some authors have switched. The talks that we have today, we start first with Professor Jiantang from Mila, who will discuss about geometric deep learning for proteins. Then we follow up with Cass, who will introduce Cass from Valence Lab, who will introduce Polaris, a new industry-led benchmark and data set. Then we have the poster session that happens at 11:15. The poster session will last 1 hour. So the poster session is presented like in 2 different parts. So half the accepted poster are presented in the first part and half in the second part. But there are still some spotlight poster here that will be presented in both poster sessions. So what we will do in between the poster sessions is that the whiteboards will be turned around so that the number 1 and 2 are presented in the first session, and number 3 and 4 are presented in the second session. For the spotlight posters, when the poster session begin, we will move them around so that there is space to see both the front and the back of the posters, and they can be presented simultaneously. So the first poster session lasts for an hour. Then there's the lunch break happening. The lunch break is also an hour, and the lunch will be provided. At 1:15, we then have the talk from Jason Hartford, who just started a professorship position at Manchester University in the UK. And he will be presenting his talk about detecting biointeraction, and a special thanks for him to fill up a last-minute role. He only learned about that a few minutes ago. Then we have property prediction in drug design and how to use that in machine learning pipelines by Raquel Rodriguez Perez from Novartis. The second poster session will start at 2:30. And finally, AlphaFold 3, which don't need any introduction to the audience here, will be presented by Max Jaderberg, who is a chief AI scientist at Isomorphic Lab. A big thank you to all the organizer. I think Johnny can take it.
There's a lot of work that went behind organizing the conference, and so I just want to say a huge thank you to, first of all, the staff at Mila, who's with the building that we're in is in Mila. So huge thank you to Carl, Desiree, Ludovic, Laurent, who are in the room, hopefully. So a lot of work went into getting things set up today. So a huge shout out to them. I also want to say a huge thank you to our group of student organizers who helped with logistics, who helped with organizing, who helped with reviewing the posters and papers as well. So this would not have been possible without their support. And also our sponsors. I want to say a huge thank you to our sponsors. Some of the representatives from each of the companies are likely in the room today. So if you can find them or you want to learn more about the companies, definitely go and ask some questions during the poster session or the lunch break as well. A few quick notes on logistics. So for the presentations today, each talk will be about 40 minutes. So we might not have a ton of time to answer questions live. If you do have questions, we have 2 microphones here in each of the aisles. Please just make your way over there and ask your questions in the microphone so that everyone can hear you. But if you don't get a chance to ask your question live, we also have a discussion space on Portal. Everyone should have received an email either late last night or early this morning that sends you a link directly to that space. Any questions that you don't get to ask live, you can leave them on there and we'll do our best to make sure that you get your questions answered. You can also scan the QR code to join that discussion space directly if you're not already on there. Wi-Fi. So there are little posters here just on the pillars. If you need to connect to Wi-Fi, the network name and the password are available for you there. We have bathrooms just around the corner here, as well as alongside the wall. As well as, yeah, that's pretty much it for questions, Wi-Fi, and bathrooms for logistics. And if you have any other questions throughout the conference, you can come and find myself or Dom, and we'll be here all day to answer any specific questions that you might have. And as Dom mentioned, you know, we have a change up in the schedule here, so we'll make sure that the website is updated accordingly. But if you don't know the schedule already, it's available on the website, and you can also scan the QR code to find that directly. Introduce Jan.
All right, so thanks again for coming here. We'll introduce the first speaker, Professor Jian Tang. So Professor Jian Tang is currently an associate professor at MILA, the Quebec Institute of Machine Learning and AI, and holds a Canadian Cypher AI chair. He's also the founder of Biogeometry, a new startup in the field of AI for protein design. His main research interests are in deep generative model, graph machine learning, and their application to drug discovery. He's an international leader in graph machine learning, and his representative work, line on node representation learning, has been cited more than 6,000 times. His group has also pioneered some work on AI for drug discovery, including the first open source machine learning framework for drug discovery, Torch Drug and Torch Proteins, that are widely used. He was also recipient of Best Paper Award at ICML and Best Paper nomination at WWW. Thanks a lot for coming.
So first, I would like to thank Dominic and the team for organizing such a great event. And it's very exciting to see the community is growing at a very fast speed. And welcome all of you to MILA. Okay, so in today's talk, I'm going to talk about what we have done in the past, like pretty much like 3 to 4 years on geometric deep learning for proteins. So I will start with the first slide to basically to tell you, okay, why protein is very important. Why do we need to work on proteins? So you know, like in living cells, most of the work are actually done by proteins, right? So you have DNA translate to RNA, DNA. So you have a DNA should be transcript to RNA and RNA translate to proteins, right? So most of the work should be done by proteins. That's why in reality, it's very important to understand the functions of proteins, which have applications in a bunch of like domains. Let's say therapeutics, right? You have antibody drugs, or in industrial applications, you have like enzymes, or you have like materials. Okay. And then so nowadays actually we got a huge opportunities for machine learning models to understand the functions of proteins, because now we got a huge amount of data sets, right? For example, for sequence data set, so we have a huge amount of like a sequence database in UniProt. So we got like I think like tens of billion protein sequence available. So that's one type of data set which is really sequence, right? And the second type of like data is really structured. So we got a lot of like PDB crystal structures in PDB, which we have like maybe around like 300,000 structures, which help us to understand protein at the atomic level, right? And this is the second type of data we can leverage to understand proteins. And the third type of data is really like experimental data. Like you know, nowadays we got like a really high throughput assays, so we can test like hundreds or thousands of proteins at a single bioassays, and then we can get the functions of each protein, right? So all these kind of data actually help us to better understand the functions protein. Okay, so in the past few years, there's a lot of work in the machine learning community for protein modeling. Of course, the easiest approach is sequence modeling, right? So you represent proteins as amino acid sequence, and then you build a protein language models, right? You just train transformers BERT on top of those unlabeled protein sequence, and then you fine tune those transformers based on some limited labeled data, right? So that's pretty much the main the most popular approach for protein modeling in the past few years. But then later we realized that okay, there's a better alternative, because you know, like for proteins, the structures determines the functions, right? And that's why in reality, so if we can better, if we can model proteins, if we can represent proteins as structures and then learn repetitions based on the structures, and that's a better approach. And that's why now people started to move from sequence modeling to structure modeling. But how do we model structures with machine learning approaches? That's we need to use geometric deep learning approaches. And that's basically the focus of this talk. Okay. So essentially we have three kinds of like research questions to answer for structure modeling. The first question is, okay, suppose now we have a 3D protein structure. How do we represent a 3D protein structure, right? Because you know, like in deep learning, we pretty much map everything to a continuous vector, right? We map a word, a sentence, an image to a vector. But here now we have a 3D protein structure. How do we map a 3D protein structure as a continuous repetition, right? And that's the first type of like a research question we have to answer. The second one is like in reality, because it's very difficult to obtain the 3D structures of molecules or proteins, right? Because we need to do like, let's say, crystallography, which is very time consuming and expensive, right? But then that's why we really need to rely on computation methods like AlphaFold 2 or AlphaFold 3 to predict the 3D structures of proteins or their interactions. And that's the second type of like a fundamental problem we need to tackle. Okay. And of course, at the end of the day, the most important problem is, okay, we always want to do generation, right? Generate models, we want to generate new proteins with good functions. Okay, we want to build diffusion models or flow matching models to design to generate new proteins. So that's basically the three fundamental questions we have been working on in my group in the past few years. So I'm gonna start with the first type of like research question. Okay, so suppose we have 3D protein structures. How do we learn the repetitions? So this is a work we published in last year's iClear. So essentially we built a geometric encoder to encode the 3D protein structures. So the input here is like, so we have like a 3D protein structure, okay? And then what do we do here? Given this 3D protein structure, we first construct a graph. So here is a graph. So this is a graph of residue. So every node here is a residue. Okay. Of course, then the problem is how do we define the edges between the residues here? So we basically mainly make use of two kinds of information. The first type of information is of course the sequence information, right? Because you know protein is still a sequence amino acids. So that's one type of information we can leverage to build the edges between the residues. The second one, of course, is really the 3D structures, right? So here, what we do is essentially we just construct k-nearest neighbor graph. So for every residue, we just connect this residue to the most the closest residues, and that's how we got the graph. Okay, of course, this graph is like not undirected; it's a directed graph, right? And then the problem of learning this protein structure repetition translate to learning the repetition of this graph, right? And then, of course, we know how to learn the repetition of graph. We just use graph neural networks, right? So we do massive parsing between the nodes or between the residues, okay? Of course, when we do massive parsing, because you know here we have multiple different type of edges between the between the nodes. So when we do massive parsing, we have to take the edge type into consideration. But anyway, so we're gonna do massive parsing between the nodes, between the residues, okay? And then once we do massive parsing, so every residue, every node has a repetition, and then we can do pooling, right? So we pull all the residue repetitions, and then we can get the repetition of the whole graph, and that's a repetition of the whole protein. So this is the first type of methods we developed, which we call a gear net. So for the gear net, essentially we are doing node level massive parsing because we pass messages between the nodes, right? But then we actually come up a better solution, which we call like edge level massive parsing. So in the second type approach, we are not doing massive parsings between the nodes. So we are actually doing massive parsing between the edges. So why do we do this? Because remember that here, this graph is not a 2D proportion graph like what we usually see. So this graph is a 3D graph, right? So there's some geometric information we haven't leveraged in this graph. For example, so the edges, we actually have angles or torsion angles between the edges, which we didn't make use in the first model. And that's why in the second model, we do massive parsing between the edges. And then when we try to do massive parsing between the edges, we actually make use of the angle information between the edges. Let me give you one example. Let's say here, the central edge we are considering here is like 4 and 7, okay? And then, so we're gonna do massive parsing between this edge, 4 and 7, between all the edges who share the ending node with the starting node of this edge. You see like, so the central edge is 4 and 7. All the neighboring edges are 2, 4, 6, 4, whose ending node is the same as the starting node of this edge, right? And when we try to do massive parsing between the two edges, as I said, we take the angle information between the two edges into consideration, and that's why we're able to better make use of the 3D geometric information in the structure. Okay, so once we do the massive parsing, so then every edge has a repetition, and then we further do pooling. So we pull all the edge repetition, and then we can get the repetition of the whole graph. Okay, so that's what we call like gear net edge, which is essentially doing like edge level massive parsing. Okay, and then for all these models, we're gonna train this encoder based on supervised data. Okay, so this is the supervised training part. And as we know, like in biology, one challenge problem is always we do not have so many label data, right? And that's why we always want to do pre-training. Okay, and that's why we come up with the second part. We're gonna pre-train this like the geometric encoder I just mentioned. So we're gonna pre-train this 3D geometric encoder based on a huge amount of like unlabeled structures. For example, the PDB structures, right? So which is really a lot, or the recent like released like a predictive structure by AlphaFold 2 or AlphaFold 3. Okay. So here for the pre-training, we actually use an idea called like contrast learning. And so this idea is very similar to what we have seen in image pre-training, right? So you know like for image pre-training, they use contrast learning. So idea is very simple. So if two patches are from the same image, then their repetition should be similar. Otherwise, their repetition should be dissimilar, right? So here we actually take a similar approach. Of course, here we have to define what does patch mean on proteins on a 3D protein structure, right? So here we actually don't use a patch. We actually use like a motif or structure motif. So here you can see, okay, given this 3D protein structure, we can extract two motifs from this protein structure. Okay, of course, actually there could be two different ways to extract a substructure or motif from this protein. So one way is just like you just take a subsequence. You just take a subsequence from the entire sequence, okay, and then you can get a subgraph, and that's one way to get the motif based on the sequence information, right? So another way is, okay, you really like get the substructure based on the spatial information. So what we do here is, okay, we just randomly sample residue on the graph, okay, and then you specify a radius, and then all the residues lie within the radius will be taken into this subgraph or into this like motif, okay? And then for these two motifs, if they are from the same protein, so their repetition should be similar. Otherwise, their repetition should be dissimilar. That's essentially the idea of like contrast learning. And then we pre-train the model based on a lot of like unlabeled protein structures, and that's the pre-training part. Okay. And then in terms of like evaluation, so we evaluate our models on a bunch of like important like protein understanding tasks, which are quite all these tasks are quite like popular and classic. For example, enzyme commission number prediction, gene ontology term prediction for the classification and reaction classification. So these are the supervised tasks to evaluate the performance of our models. And then we also have a pre-training data set. So one way to pre-train is really like we pre-train on the PDB structures, which are experimental structures, which are more accurate. But you can also pre-train the models on the predicted structure by AlphaFold or AlphaFold 2. Okay, so that's the pre-training part. So we can take a look at some of the results. So we have two kinds of evaluations. So in the first evaluation, it's pretty much like supervised learning. So we want to evaluate the performance of different encoders. So the first type of method is essentially like sequence-based methods, because like we represent proteins as a sequence. So we have different sequence encoders like traditional LSTM or transformer, right? And then we can also represent proteins as graphs, okay? So which is the second type of like methods. And then the third type of methods is a method we propose, which we represent proteins as 3D structures, and we train a 3D geometric encoder. So in general, you can see that like for the supervised learning part, of course, our methods can get better performance because we actually make use of the entire 3D structure information compared to the sequence-based and also the graph-based methods, which is not very surprising, right? For the supervised training part, okay? And then you also see even on the if you compare the performance between GeoNet, which is doing massive passing between the nodes, and also GeoNet Edge, which is doing massive passing between the edges, you can see that. So GeoNet Edge, the performance is much better than the GeoNet. So because for the GeoNet Edge, we are doing massive passing between the edges, which is better, which is able to better make use of the geometric information in the 3D structures. So that's the first part, which is supervised learning. Okay, and then in the second part, of course, we care about like pre-training. We can pre-train the model on labeled protein structure, can first improve the performance. So here we just show one result. You can see that for this multi-view contrast is based on the GeoNet Edge encoder. So you can see that. So after pre-training, the performance can be further improved, showing that like a pre-training can indeed help improve the model performance. So that's some experimental results. Okay. And then so here we also did some study on the sensitivity of pre-training on different data sets, because we can actually pre-train the models either on the PDB database, which the structures are obtained through experiments, right, which are more accurate. But then we can also pre-train the models on the structures predicted by AlphaFold 2, which of course is less accurate because it's obtained through prediction. But of course we have many structures because we can get as many as we want by running AlphaFold 2 on the protein sequence, right? So in general, we see that actually either pre-train the models on the PDB database or the AlphaFold predict database, we can get similar performance, showing that actually our pre-training methods is actually quite robust, either training on the real world experimental structures or the predicted structures. Okay, so that's an aberration study on pre-training. Okay. So this is the first type of like research we have been doing, which is really doing pre-training on 3D structures and then fine-tuning the models on some limited downstream supervised data set. Okay, and then we actually apply this methodology to a real-world application, which is called like antibody optimization. So antibodies is kind of like biologics, right? So in drug discovery, usually, so let's say antibody drug development. So we usually got an initial antibody through animal immunization. So you do animal immunization and then you can get some antibody candidate. But usually the antibodies we got through animal immunizations, sometimes they are not ideal. For example, the binding affinity is not like strong enough. And then we need to do some optimization. We need to improve the binding affinity. Specifically here, if you look at this example, so we have a drug target called the CAL, and then this is an antibody bind to this antigen. And then what we want to do here is, okay, we want to optimize this antibody to increase the binding affinity between the two. Okay, so how do we do this? So in reality, what we do is like we just want to make some mutations. We want to make some mutations on the interface between this antibody and this antigen. So we want to make some mutations. Most of the time, we make mutations on the CDR loops. We make like a few mutations to improve the binding affinity. Okay, so essentially, what we want to here, what we want to do here is we actually want to build a machine learning model. We want to predict how the binding affinity changes if we make some mutations on the antibodies. So we want to build a predicting model on called like delta delta G, which is a binding affinity change. As we know, like for delta delta G is actually the difference between the binding affinity of the mutants and also the binding affinity of the wild type antibody. So that's the definition of our problem. Okay, and then for this problem, so we also actually use a similar approach. We also do we also do like geometric pre-training and also fine tuning. Okay, so here we have like a pre-training model which is pre-trained on the 3D on labeled 3D protein structures, and then so we also have like a supervised learning model which is called like gear bind. For this gear bind model, so our goal is trying to predict the binding affinity change delta delta G. Okay, so once we have this like binding affinity change prediction model, okay, and then in reality, given an antigen and antibody complex, so we're gonna do in silico screening based on this binding affinity change model, right? Because we're gonna measure the binding affinity. We're gonna measure the binding affinity change for each possible mutation, right? And then we're gonna rank the mutations according to the binding affinity change. We're gonna keep the ones with optimal binding affinity improvement, right? And then that's how we can get some mutant candidates. Okay, and then once we do this, of course, this is still done in the in silico way, right? And then what we do is we're gonna further test those candidates in real world wet labs. Okay, so mainly like binding assays. So for example, we can measure like scores like ELISA or SPR. Okay, and that's how we can really got the true binding affinity change. Okay, so usually in the first round, we only like do some single mutations. Okay, and then after finish the first round of like experiments, and then you can see, okay, which single mutations is good. Okay, you can see the binding affinity change for different single mutations. So after the first round, and then we take the optimal single mutations, and then in the second round, you combine the best single mutations, and then you can first test the double mutations or the triple mutations, and that's how it works in practice. Okay, and there's a general framework of antibody optimization. So we can look at some details. Okay, so first the supervised learning part. As I said, in this task, our goal is trying to build a machine learning model to predict the binding affinity change, which is essentially delta delta G, right? So for delta delta G, as we know, like is a difference between the binding affinity of the mutant and also the binding affinity of the wild type. Okay, so what we do here is like it's very similar to what we do before. Okay, so here's the structure of the wild type, which is antigen antibody complex structure, and then we're gonna construct a graph out of this complex. But remember that because here we only care about like binding affinity. So for binding affinity, it mainly depends on the interface between the two molecules. And that's why when we try to construct a graph, here the graph is only constructed based on the interface of the two molecules. Okay, and then so we're gonna construct a graph based on the interface. And then once we have this graph, okay, remember this graph is not 2D. This graph is still like a 3D graph. So we're gonna do massive passing on the graph. So we can do different level of massive passing. So we can do residue level massive passing. We can do atom level massive passing. So we can also do massive passing between the edges, right? Which is able to make use of the angle information between the edges. Okay. So once we do the massive passing, so we can do pooling, and then we can get the repetition of this complex structure. And that's what we do for the wild type. So we can also do the same thing for the mutant, right? So we do the we construct the graph and also do the messy passing for the on the graph, and also then we can get repetition for the mutant, and then we take these two as input to predict the final binding affinity change, which is delta G, right? And this is our machine learning predictive model. So this is a trend based on some supervised label data. Okay, so this is supervised training. Okay, and as we just said, we're also gonna do like pre-training. Okay, so we also pre-train the model on some unlabeled protein structures. Okay, so here we actually also use a contrast learning idea. Okay, so here we have like a protein in a complex structure. Okay, and then so we also construct a graph based on the interface, and then we do geometric like deep learning. We can get repetition of this interface. Okay, so we do this for the Y type, and then we also do this like for the mutant. We can get a repetition for the mutant, and then for contrast learning, we just train a classifier to tell whether this is a true structure or a mutant, right? Okay, of course you may have a question. In reality, how do you get the mutant? So what we do here is like, okay, given the real world protein complex structure, we just randomly mutate some residues on the interface. Okay, and then for each mutated residue, because you also need to determine the structure of the side chains, right? So how do we handle the structure of the side chains? So here we use a very simple approach. We just sample the side chain torsion angles from a Rotamer library. So it's not very accurate, but it's a quick way, and also still a reasonable way to get the structures of the mutants. And that's how we calculate the repetition of the mutant structures. And then as we just said, we're gonna train a binary classifier or contrast learning to pre-train the 3D geometric encoder. Okay. And then so we do some in silico evaluations mainly on the ScanPy data set. So here we actually compare different kinds of methods. So the first two methods, FoldX and FlexDDG, so they are physics-based methods. So FlexDDG is based on the Rosetta energy function. Okay. And then the third method, bind DDG, is a deep learning method. Okay, and then our gear bind approach, and also the one with pre-training. So you can see that in general now on this task you can see that the deep learning method can significantly outperform the physics-based method, showing that nowadays, as long as we have enough data, so the knowledge we learn from the data can outperform the physics-based energy function. Okay, and then for our gear bind model, you can see after doing pre-training, so you can see that the performance further improves, showing that pre-training can indeed help the performance. And then we also do some ensemble. We actually ensemble all the physics-based methods and also the deep learning-based method. So you can see that after ensemble, so nowadays, like on this task, the Spearman correlation can achieve as high as 0.6. I think the accuracy is already good enough, can be used in practice. Okay, and that's why in reality, actually we test the performance of our models on real world examples. So here, this one is a real world like antibody drug, which is called like CR3022. So this is like SARS-CoV-2 antibody, okay, which is obtained through from the patients. Okay, so we also know the complex between this and antibody and also the antigen. So here the antigen is the RBD domain of the SARS-CoV-2. Okay, so what we want to do here is like we want to make some mutations on the CDR loops of this antibody to improve the binding affinity. Okay, so we do some in silico screening to get which mutation can improve the binding affinity. So in the first round, we only identify single mutations because in our like label data set, we only have the binding affinity change labels for single mutations. That's why like for the predictive models, we only do single mutations. We don't have the label data for double or triple mutations. Okay. So in the first round, we pick like I think 8 optimal single mutations, and also we also take the best 2 single mutations as double like mutations, And then what we do is like we test all these like mutations or antibody sequence in the wet lab. Okay, so you can see that like in the first round, this one is wild type, this one is wild type. Okay, and the others are mutants. Okay, so in general, if the curve is like on the left top corner, the performance, the binding affinity is good. Okay, so in general, you can see that for most of the mutants predicted by our model, the binding affinity actually improves compared to the wild type. So this is in the first round. Okay, so in the first round, and then we can identify like a few good like single mutations, and then in the second round, we're gonna combine those like optimal single mutations. We're gonna test like double mutations and also triple mutations. Okay, so in the second round, eventually we found like molecules with 3 mutations, and the binding affinity improves like 17-fold compared to the wild type. So I want to emphasize, actually, this problem is already is very challenging because for the original molecule like CR3022, so the binding affinity is already very high. It's like 1 nanomole, and now we increase the binding affinity to like 6 to 10⁻11. So this is actually pretty challenging in practice. Okay. And then, so all these like experiments, like, so usually for every round of experiments, so this can be done in 1 week. So that's, so in total, we finish finish the computational and also wet lab experiments within 2 weeks. Okay, and that's pretty fast. You know, traditionally, how people tackle this problem. So what they do is like, okay, they're gonna like random mutate the residues, and then they're gonna build a library to do the screening. So traditionally, usually this take like 3 or 4 months, and now we actually do this like within two weeks, which is very efficient. Okay. And this is the first research questions, like how do we learn, how do we pre-train protein repetitions and also use those repetitions for different applications. Okay, let me see how much time I have. Still have 10 minutes. Okay. For the second problem, we're gonna talk about like how do we like predict protein structures based on sequence. So we already have a lot of great work from DeepMind, like for example, AlphaFold 2, ESMFold, and also the recent AlphaFold 3. Okay, so in our group, we have been also working on this problem. Okay, but you know, like for AlphaFold 2 and also AlphaFold 3, I think maybe many AlphaFold 2. So they mainly actually work on predicting the backbone structures. So they actually didn't do well on predicting the side chains of the proteins, and that's why we have been actually trying to working on building better machine learning models to predict the side chains of proteins. Okay. And sidechain predictions is very important for problems like protein-protein interactions or protein-ligand interactions, because usually for two proteins, when they interact, they usually mainly interact through those side chains. Okay, and then for traditional approaches, we have methods like sampling from Rotamer libraries, or you can do sampling from some physics-based energy functions, for example, like Rosetta 2. But those like physics-based energy functions, there are two limitations. First, they are not accurate, and also the sampling process is quite slow. Okay, and that's why we're also trying to define or develop diffusion models for sidechain prediction. For sidechain prediction, essentially we want to determine the coordinates, the structures of the sidechains of the proteins. And then, so one way to do this with diffusion models is essentially you can try to build the diffusion models on the Cartesians on the 3D coordinates of the atoms, right? And then in this work, we actually did it in a different way. So we actually didn't build diffusion models on the Cartesian space. We actually built the diffusion models in the torsion space, because you know, like for protein, for the side chains, so the degree of freedom of the side chains is not the coordinates of the atoms. The degree of freedom of the side chain is controlled by what? By the torsion angles, right? And that's why here we actually define a diffusion process in the torsion space instead of the Cartesian space. Okay. So specifically, we actually propose an autoregressive diffusion models. So you know, usually like for the side chains of proteins, we have like we have 4 torsion angles. So here we actually do this like in an autoregressive way. So we first like this is similar to what like ChatGPT is doing, right? So we first try to predict the first torsion angle, and once we know the first torsion angle, we condition on the first torsion angle. We predict the second torsion angle. We do this like autoregressively. Okay. Of course, you may ask, why don't we like predict all the 4 torsion angles simultaneously? We tried that. Actually, it doesn't work that well because so in reality, we found that actually the four torsion angles are very highly like interdependent. It makes the model very difficult to learn the dependency of the 4 torsion angles. That's why we found actually doing this like autoregressively make the learning process much easier. Okay. And then so we compare we evaluate the models on the CASP13 on the proteins in the CASP13 competition. Okay, so we compare our models with traditional like physics-based methods and also recent deep learning based methods. So our model can outperform all those like traditional physics-based methods and also the recent deep learning based methods, but with much like less parameters. Okay, I'm not going to get into details due to time limitation. Okay. And then so this is like predicting the side chains of proteins. And this is another work we did recently, because like for AlphaFold 2, one limitation is that for each protein sequence, we only predict one structure, and this is not true, right? Because in reality, actually, a protein can have multiple stable conformations, right? So essentially, so what we want to do here is, suppose this is the energy landscape of the protein structures. What we want to do is we actually want to sample. We actually want to sample from this energy landscape, right? So we actually want to get the structures with minimal energy, okay? And that's what we want to do. 5 minutes, okay? And then so there were a lot of like work in traditional biophysical domain. For example, people use like Monte Carlo or monic dynamic methods to sample from either an energy function or a force field. But for those methods, there are 2 limitations. The first limitation is of course, it's very computation expensive, and that's one big limitation, right? And the second one is usually most of the time they can be, those like sampling methods can be easily get trapped by some high energy modes, right? In other words, they cannot cover different modes of the energy function. Okay, and these are some traditional approaches. And recently, there are also some work. People have been trying to build a generative model from the molecular dynamic simulation data. So what people do is, okay, they first do some short run of molecular dynamics, and then they can get some training data, right? And then they fit a generative model to those like MD generated data, okay? And then there are also some limitations for this kind of approach. First, it's very expensive to get the MD simulation data, right? Because for every protein, so we have to do some rounds of MD simulation to get data, and then we will train a generative model, right? And remember that this kind of generative model is only specific to that protein. You cannot generalize this generative model to another protein. In other words, suppose you have a new protein, so you need to do this again, right? You need to do the MD simulation again, and you need to do train a generative model again, and that's the limitation of this kind of approach. So what we did here in this paper, this paper we published in this is iClear. So we train a generative structure translation model, not from the MD generated data. We actually train this model from the crystal structures from PDB essentially. Okay, so our approach is somehow actually inspired by traditional like approach called like a simulated annealing in MD simulation. So in those in the simulated annealing approach, so what did they do is like, so they have some like exploration phase. Okay, so you see the red one. Okay, so essentially what we do here is they are doing like exploration, and then locally they're gonna do some annealing. Essentially, they're gonna find the local minimal. They're gonna find the structure with local minimal energy, right? So here, what we do here is we're gonna start from a protein conformation. For example, the conformation can be predicted by AlphaFold 2. Okay, and then once we have a starting point, we're gonna do exploration. So how do we do the exploration? So here we actually just do some stochastic perturbation. So we do some stochastic perturbation so we can cover different space of the energy landscape. Okay. And then once we do this, okay, we're gonna use our like generative model. Essentially, the score we train a score function. So we use that to map the perturb structure to the local minimum, right? So we can do this traditionally using MD simulation, which is very slow. But now we do this using diffusion models, right? And that's what, because we have trained like such a structure to structure translation model, right? So we have like a noise, we have some noise structure, and then we try to map this noise structure to a stable structure with low energy. Okay, and this is based on the actually the IPA architecture in AlphaFold 2. So I'm not going to go into the details. Okay. And then finally, so we've been working on like diffusion models for protein design. So I'm not going to the details because of time. And then so in my startup, besides working on the building the generative models or machine learning models, we also have built up our web labs so that like within like 2 or 3 months, 2 to 3 weeks, we can test like hundreds or even 1,000 proteins, and then we can get the labels of those proteins. We know, okay, what is the binding affinity, what is the functions of those proteins, and those label data is gonna be feedback to our machine learning models. We're gonna fine tune the generative models based on the label data and do another round of like design. Okay, so that's and that's very exciting. And so we are like designing different kinds of proteins like antibodies, enzymes, in different applications. Okay, so with that, I will stop here. Thank you.
Thanks a lot for the great talk. It's really interesting to learn about all the generative work that you've done for protein and representation work, and also to see that it works in real wet lab experiments. So yeah, that's I think it's a good sign that like all the work that everyone here is doing like will have an impact in drug discovery. We have time like 2 minutes for questions. So for each talk lasts roughly 40 minutes. And we have like a few minutes in between the talks only for 1 or 2 questions. For the questions, like whenever you have them, you can line up at the microphone. But usually we rather keep them after the talk. So go ahead.
Hi, so it's very interesting. The protein problem is very interesting because you have to make a choice about resolution, what you're going to model as the nodes of the graph. So for example, you could have had higher resolution, the nodes of the graph could have been atoms instead of residues.
Yeah.
You could have had lower resolution, you could have modeled an alpha helix as a rod.
Yeah.
So how do you make that decision?
Yeah, that's a good question. I think ideally you probably want to ideally you want to represent the proteins at the atom level, right? That's ideally okay. But usually there are a few challenges, because if you represent proteins as atom level, then your graph is going to be huge, right? So because then you probably have like 1,000 atoms in a graph, and that's very computation expensive, and that's one thing. And the second one is like, if you represent proteins as atom graph, it's going to be very sensitive, because sometimes like you may not be able to get a very accurate protein structure, right? If you have an experimental structure, that's fine, but sometimes like even the experimental structure is not accurate, right? Most of the time you can only get the structure by running AlphaFold 2, right? Which those the side chains are not accurate. So I think so there are some limitations on as I said, ideally atom graph is optimal, right? But there are some challenges. But in some cases, I think at atom level is actually quite helpful. If you see in our case, when we try to model protein-protein interaction, we actually don't care about the entire protein structure. We only care about the interface. So in that case, we can only take the interface part, and then the graph is very small, right? Okay, and that's one thing. Okay, and then so in reality, I think it's good like you combine different resolutions, atom level and the residual level. So residual level, yeah, is the good thing is that it's less accurate, but it's more robust, right? Yeah. So, and that's what we found. You always like combine the two is always ideal. Yeah.
Great, thank you.
Thank you for the great talk. I might have missed this, but like can you hear me? I might have missed this, but like in your second part of your talk, when you present the string to string method, like
Yeah. So you mean training?
how did you address the sidechain conformational sampling problem? I might have.
Right. So I think we actually do an approximation. We first sample the backbone structure, and then given the backbone structure, we actually use some Rotema library. It's not 100. It's not that accurate actually. So we actually do some approximation there. So we actually mainly train a general model to sample the protein structures and the backbone structures. And once we have sampled the backbone structure, we condition the backbone structures. We use some Rotema libraries to quickly to sample the sidechains. It's not that accurate, but it's a relatively efficient way and accurate way. Yeah.
What's the Rotema library you mentioned?
So Rotema library is some kind of like so like people for different torsion angle for each torsion angle there's some distribution. It's not totally like a random. So people got that statistics from the PDB database. So basically, for every torsion angles, you have some statistics from the PDB database. And then what people do is you're gonna draw a sample from that distribution. That distribution is got from the PDB database.
Okay.
That maybe is a intuitive like explanation. Yeah.
Thank you.
Yeah.
Right.
Hi.
Let's make this the last question.
So my question is about the contrastive loss you're using for the 3D geometric and structure encoder.
Yeah.
So you mentioned that the positive pairs are structural motifs from the same protein.
Yes.
And the negative pairs are the one from different proteins.
Yeah.
But I wonder like how you make your training set then, because sometimes you may have like proteins that are very structurally similar in the same like.
Yeah.
Yeah. So what if you pick motifs from one structure, but then the negative would be like a protein with very similar?
Right. So I think there's always a problem like this, like in contrast learning, even in the case of like image repetition, right? So you take two patches from the same image, they may not be similar, right? And the two patches from different image, they may be similar, right? So, but I think this is more like a signal versus noise ratio. It's more likely to happen, right? Patches or motifs from the same proteins, they are more likely to be similar than the ones from different proteins. So I think it's more like from the statistical point of view, or signal versus noise ratio. Okay. On some specific cases, it doesn't hold for sure. Yeah, but the good thing is that for machine learning models, that we can learn from the noise data, right? Yeah.
Thanks again. A big round of applause for Professor Jianpeng. All right, so we'll move to our next speaker. Yeah, the slides are already there. So I'd like to present you Cas Wognum, who will present the work Polaris, an industry-led initiative to critically assess machine learning for real-world drug discovery. Cas is a machine learning engineer at Valence Labs. Within Valence, he has contributed to several open source projects in the data model ecosystem and the different toolkits, which led to the Polaris project, which he will present today. He holds a master's degree in artificial intelligence from the University of Utrecht in the Netherlands.
Yes. All right. Good morning, everyone, and thank you for the introduction, Tom. So today I'll be presenting, I'm actually very excited to present Polaris, which is an industry-led initiative to critically assess machine learning in drug discovery. And we'll unpack what all of that means in the next 30 minutes or so. But before we do, or to get us started, I would like to give a bit of an introduction as to the challenges we face when we're trying to benchmark machine learning models within drug discovery. So I think we can all agree that benchmarks play a quite important role within machine learning research. That's broadly applicable. That's not just drug discovery. I would say that they don't just provide us with a way to measure performance or to track progress. I would say they actually also inspire entire new research directions or even unlock breakthrough results. I would say that the gold standard within evaluating or within machine learning benchmarks is a competition. What do I mean with a competition? I mean to say that the data that is used to evaluate the model is ideally like newly generated and unknown to the people that are modeling that data. And I think within drug discovery, the goal, like one of the most famous examples is perhaps CASP, which is this competition for predicting the protein structure. I think most well known because it was AlphaFold that submitted their model, their predictions to that specific competition. However, one thing I think is worth highlighting is that CASP wasn't a one-time event. It's actually I think it's a lot of the value comes from the organizers quite rigorously, difficult word, trying to identify the most valuable innovations within the community with a certain like regular interval. Yes. Okay, so we know how to do benchmarking really, really well, right? We can do these competitions. So why don't we just do more of that? And the answer is unfortunately quite simple. It's just data generation is expensive. So I'm putting some numbers on the slides with certain assays, how much it costs to test for a single molecule. And you'll just quickly see that it's not feasible to run competitions with new data with a certain interval, especially given how high the pace of innovation is within the machine learning community. So we need something else than competitions. And what can that be? That's where a static benchmark comes into the picture. So in a static benchmark, different from a competition, the test set is known. You're taking some data that is publicly available, you split it in a train and test set, and then you train on the train set, evaluate on the test set. Unfortunately, there's some issues with static benchmarks. Results, like the evaluation on a static benchmark, given that the test set is known, is more likely to be biased. This can be due to test set leakage, but it's also just because the incentive is there to do really well on this test set. So even sort of like without bad intentions, you might find yourself training like 100 different models, evaluating them all on the same test set and just picking the one that does best. But the question is that still like a valuable innovation at that point? This is a problem, I think, for all of machine learning. This is not just drug discovery, and I think a lot of us are familiar with this challenge. I think one additional point that's worth making is that these challenges are further amplified within drug discovery. Within drug discovery, I think the biggest like differentiator, or within ML for science, perhaps more broadly, is that the data that we're working with is no longer intuitive, right? If I train a computer vision model, I can look at some of its predictions and I can do a pretty good job without any formal training, simply because I'm human, to understand whether that model is doing well or not. I cannot do this within drug discovery. I think a lot of people, like chemistry, biology, physics, these are all fields that people do their entire PhDs on, and they just understand a small portion of it. And the data is just less intuitive to work with. There's other factors, such as that the data is more variable, which we'll get into in a bit. But I just want to make the point that I think these challenges are further amplified within drug discovery. Okay, so what sort of situation is this creating? So I'm actually a computer scientist by training. I'm not a biologist or a chemist. I actually think I wish I would have done more of that in high school, but I didn't. So what happened when I first joined and first started applying my machine learning skills to drug discovery? Well, I did everything I was taught to do, right? I selected a data set, I split my data set. I actually tried to make very informed choices as to how to split the data set. I did hyperparameter search and all of the above. And at the end of that sort of trajectory, I had a 1% improvement over the state of the art. Hooray, we solved drug discovery, right? No, of course not. It's unfortunately not that easy. because then you bring those results to your colleagues who have a more of a background in chemistry or biology or drug discovery in general, and they'll start asking you all these like annoying questions. What's the statistical significance of the results, right? What's the applicability domain of your model? Those compounds you generated, they look great, but there's no way we can synthesize them. How did you handle stereochemistry during dataset curation? Could you actually explain the predictions to me? I just want to make sure we're doing the right thing here. What's the decisional impact? And even questions that maybe as a computer scientist sound simple, turn out to come up quite a lot, right? Like, how do I actually install your model? How do I use your model? So the importance, well, one thing maybe worth saying, like, I don't expect you to know what all of this means, but I think that's precisely the point, right? Like, as a machine learning scientist, you're not trained to think about these things from the get-go, and you might have picked up on it here and there, but you're not really aware of the implications of all these questions. I think given the importance of benchmarks within the machine learning community and pairing that with the inherent difficulty of proposing a benchmark, this is leading to a situation where there is a growing gap between perceived progress and actual real-world impact. And that's something that we set out to address. So everything I've said so far is perhaps a bit on a high level, right? I've tried to convince you that machine learning in drug discovery is difficult. I would like to illustrate that with some examples just to bring the point across. So first things first, if you want to benchmark your machine learning models, you'll need a data set. And data set is, like, I would say in drug discovery, there's quite a bit of data publicly available within databases such as Campbell. But this data is very heterogeneous, it's imbalanced, and it's subject to experimental noise. So to actually curate a data set from such large public databases, you need to very well understand the underlying data generation process and the underlying data modalities. There's this interesting, it was first a blog post by Greg Landrum and then turned into a paper. If you don't know Greg Landrum, he's one of the main contributors or the main maintainer of RDKit. He has a really nice blog post worth reading. But one of the things he did is he did a simple experiment, like, okay, if I just aggregate all the data from these public databases in the most simple way, doing some sort of sanity checks, but not looking into too much of the details, what's the effect of that? And he found that it actually led to quite noisy data sets. So you can see that on the top left. He then came up with a way to improve the automatic data creation by having more stringent like filtering criteria. And he definitely managed to improve this, the situation, as we can see in the bottom, sort of the bottom row. However, one thing that's worth noting in this particular assay, the IC50 assay, the experimental noise of the assay itself is normally estimated about 0.3 delta on this axis here I'm showing. So we can see that even though with this automated curation, he managed to sort of reduce the variance between replicates, he's still above the experimental noise. So we can still expect there to be some noise in this data set. Okay, so what do you need to do if you want to improve things even further, or maybe what should you do nonetheless? And there was this other interesting figure within our space, Pat Walters, and he also writes a great blog. If you haven't heard, I highly recommend it. And he actually wrote a quite opinionated blog post on some of the things you should do to sort of further curate down your data set and make sure you have a high quality data set. Okay, so that's the data. Then the next question is, okay, now we have this data, how do we turn that into a machine learning task, and how do we evaluate performance on that machine learning task? So that is the questions such as, how do I split my data set, or what metrics should I use to evaluate performance? And here again, you need you need a good understanding of the downstream application you're trying to model in order to make the right choices in how to split your data and how to evaluate which metrics to use to evaluate performance, right? So if we think about splitting, I think a lot of people in the room will know that it's probably a good idea to use a scaffold split rather than a random split, because that's a more difficult splitting method. However, it's actually not that straightforward, right? Like, if you use the Mirko scaffold, there was another nice blog post by Greg Landrum again, and he showed if you use the Mirko scaffold, that doesn't really align with the intuition that humans have as to what a scaffold is. So what you end up doing is actually you find that there's very few molecules per scaffold if you do it in this automated way. And it ends up being a little bit more like a random split rather than a completely like scaffold split. Now we can even go one step further and ask ourselves, okay, we know that the scaffold split is more difficult than the random split, but is it difficult enough? Or is it perhaps too difficult? And here it really comes down to, again, your downstream application. We wrote this paper at Valence on molecular out of distribution where we tried addressing this challenge. I don't think we found the perfect answer, but if you're interested in sort of learning about some of the considerations there, I recommend giving it a look. So all of this to say, like, also, like, even if you have a high quality data set, formulating a task that is representative of real world drug discovery remains a challenge. Okay, and the final thing I want to just quickly bring up is the difficulty of method comparison. So now that we have our performance metrics, how do we actually tell whether one method is better than the other? And what we're used to seeing is these, or maybe that's a bit of an oversimplification, but one of the ways in people can present the result is with these tables, right? These leaderboard-like tables, where in bold you get the method that did best. I think one thing that's worth highlighting here is that machine learning model, or like any data-driven model really, is subject to some forms of uncertainty, right? I think an interesting paper there is torch.manual seed 3407 is all you need. This just showed that if you just change the seed of your model before you start training, you can actually see quite a big variance of performance in the final results. Now, besides introducing uncertainty in the model architecture itself through the seed, there's also a lot of uncertainty that's coming from the data, right? If I do a specific split and then I like if I do cross-fold validation and I train my model on multiple splits, you'll see that the results are not equal across all these different splits. Actually, even more so, because drug discovery, the data is so heterogeneous and imbalanced and noisy, you'll find that the variability is even larger. So you should be using some sort of way to sample performance for different splits and different seeds. Cross-validation is a good option here. You should also be using appropriate statistical tests to then compare those distributions. But again, I think this just nowadays isn't really, well, I'm not sure. I'm curious to hear your thoughts afterwards, but like this nowadays is not really being thought anymore within the typical machine learning paradigm, because if we're comparing it to computer vision, data sets there are so large that any result can be assumed to be statistically significant. But here within drug discovery, this becomes all the more important again. And actually, I think within computer vision, it's important too, but that's another topic. Okay, so those are some of the challenges that we observed. So then we thought, how could we go about addressing some of these challenges, or at least making an effort to? And what we came up with was Polaris. So Polaris is a industry-led initiative, and the goal of this initiative is to close the gap between perceived performance and actual impact. I think it's worth highlighting that what we're not doing is we're not proposing one specific benchmark or a set of benchmarks that we have carefully curated and that we've tried to popularize among the community. I think what we're rather trying to do is we're trying to lay the foundation where other people, where the entire community can then start building upon to work with more high quality data sets, work with more high quality results, high quality benchmarks. So specifically, what does that mean? We're working on three things. First and foremost, we're working on a recommended set of benchmarks and data sets. So of all the This can mean two things. So like of all the benchmarks and data sets that have already been proposed within our community, which ones of these are of high quality and should you be using? But this can also mean perhaps that there is a need for additional data generation. Perhaps we don't have the data to solve a particular problem, and then that's something we're interested in exploring as well. Second is that we want to have guidelines for dataset curation, model evaluation, and model comparison. Here, the goal is really to make these guidelines very beginner friendly, so that independent of your background, you can just read these papers and you have a good understanding of some of the best practices, such that it's easier to do the right thing. And finally, we're also working on open source software. So the thinking here was, we can write the most fantastic paper and publish it in Nature or whatnot. But we were worried that if we didn't actually follow up with it, make it easy to implement these guidelines, that people would not be inclined to use them. So we want to make sure that there's software to simplify the adoption. I think it's finally, I think it's worth saying that we have this grand vision of doing this for all of drug discovery, of all of machine learning based drug discovery. That's a big challenge just to get started and to sort of see the format that we can use to structure this collaboration. We're focusing initially on small molecule predictive modeling. This is not something we're doing alone. I don't think we have all the answers. I don't think any company has all the answers. So that's why we made a very specific effort to work together with some of the key opinion leaders within our field. And you can see they're all from different companies as well. I think that's important. And one final thing I wanna just highlight is this beautiful quote: "Don't let the perfect be the enemy of the good." I don't think we will have all the answers. We'll do our best to come up with sort of like a good first pass. But ultimately, I think this really needs to be a community-run initiative. We need to sort of, everyone needs to chime in and share their opinions. So I don't think we will be in a position where on day one, we'll have solved this problem. What we're rather trying to do is just work towards, like, just make some progress, try to improve the state of the art. And I think there's some low-hanging fruits that we can already address right now. Okay. Then finally, I want to just quickly touch upon what we have been calling the hub. So if you remember this third bullet, we are working on this open source software to simplify the adoption of these guidelines that we come up with. And that open source software will be centered around the hub. So what's the hub? The hub is a benchmarking platform. So think of like Kaggle or Hugging Face. However, we've built it from scratch specifically to address some of the challenges that we're facing in drug discovery. The mission is to make it easy to do the right thing and wrong to do the hard thing, right? So if you're using this platform, whatever guidelines or standards we come up with, we want to make it as easy as possible to implement those standards in your research by default. In the current state, so that's the mission, that's where we're working towards, but this is like an ongoing process. In its current state, I would say it best described as like a source of truth. So we've built out this platform to host the wide variety of data sets and benchmarks you might encounter in drug discovery. And if you use our platform, you'll be absolutely sure that the data set you're using like this week will be the same, it will be the exact same data set as if you access that same data set in another month from now. Like we want to make sure that there's sort of that we're operating as a source of truth. So this platform is built to host a wide variety of data sets in drug discovery. So whether you're working in phenomics, or you're working with proteins, or you're working with conformers, or you're working with just 2D molecules, or anything else you can come up with, we aim to support it. The way we're thinking about this is, all these different modalities have their dedicated file format. So we were like, there's a lot of complexity in supporting all of that. However, we're really building this platform to be a machine learning ready platform. So what we're thinking is, independent of whatever modality you're using, at the end of it, you'll have to convert it to some sort of ND array as input to your machine learning model. So our platform uses some cool technology to host ND arrays, essentially. We're also making we're doing our very best to make the barrier of entry as low as possible. So if you have data that is currently saved in an SDF file, we can automatically convert it to something that's compatible within Polaris. And finally, not finally, but next, we are also, we're supporting the wide variety of dataset sizes that you encounter in drug discovery as well. So if you're working in small molecule drug discovery, you might be used to only working with a couple of kilobytes, maybe megabytes. However, if you're working in phenomics, datasets can get very large. So there's one that Recursion open sourced, which was about 83 terabytes. And we're aiming to support like everything within that range. And finally, we also put a lot of effort in making sure that out of the box, you have quite performant data access. So whether you use this largest data sets of 83 terabytes, and you load the data directly from the cloud, or you're using a small data set, we made a lot of optimizations to make sure that sort of the out-of-the-box data loader is fast and can be used within machine learning. Okay, so all of what I've been saying so far is no longer, well, still a prototype, but it's not just in my head. It actually exists, and this is the API that we ended up with. The cool thing is that this API will be the same, independent of what your underlying data set is. So that hopefully makes it also very easy to transfer your skills from one domain to the next. Okay, so what's then next? So the current state is this, where we built this sort of like general purpose platform, which really operates as a source of truth. We're already working on a couple other things. So one thing we're working on is competitions. Right now, whenever we evaluate predictions, we do that on your local machine, which means that we need the client can get access to the test set if they really wanted to. We're working on a scenario where that's no longer needed. So you actually upload your predictions to our server. We'll give you back the results. This is just to make the evaluation less biased, but also to support hosting competitions that people are running within the community. Another thing we're working on is data visualization. So if you upload a data set, we can automatically visualize the molecules that are in it, for example. And another thing we're working on is review or expert review. So, right, like we're coming up with these guidelines. One thing I should have said earlier, but this platform is going to be community run. So anyone here that wants to upload a data set, you can. That does lead to a bit of an interesting case where, okay, if anyone can, how do we make sure that people can still find the resources that are the highest quality? And that's where this expert review sort of comes in. So you can, in a simple format, you can think of this as a comment feature on each data set where people can leave comments saying like, hey, I'm not sure about this, perhaps try changing it, or this seems applicable in this scenario, but not in this scenario. In a more complex scenario, we're thinking of working together with experts within our domain to actually evaluate the data sets and benchmarks, a bit similar to like how you would do that for like peer review within the academic community. And then the question is, what else? And that actually leads me to my final slide. So this platform, like I was saying, it exists, it's live. And if you, we're happy to give everyone here like early access. We just ask you to follow the Twitter account, Polaris HQ. You'll get a link, you can sign in, you can start using it from there on out. And the question really is like, we've built this platform out to sort of like a minimum that we think is needed. And now we're really interested in learning what other features would you like, right? Like what works, what doesn't work, what assumptions we made make sense, what assumptions we made don't make sense. So that's where we're really interested in hearing all of your feedback. And yeah, with that, thank you everyone for listening. And if there's any questions, I'm happy to take them.
Yeah, thanks a lot for the great talk. Like it's really interesting to learn about Polaris, and I can't wait to see the outcome of those big competitions. As a reminder, like you can line up at the mics if you have questions. So we are a bit in advance on schedule. Like don't hesitate. We'll have time for a few more questions. After that, we'll be heading to the poster session. Thank you.
Hi, thanks for the great talk. I wanted to ask, like, is there there's a lack of like relative benchmarking in which we are creating data sets, not just one single data set, but multiple data sets with known signal to target ratios across them, which helps get a lot of the noise sort of similar across each of them. And I was wondering if that's something you knew as to like why we don't do much of that right now. Is that because we are still getting a hang of benchmarking in general?
Yeah, so your question is, let me try repeat it to see if I understand. So the question is, there seems to be a better way in which we could generate data sets for benchmarking, but we're not really using that as of now, right? Yeah, so I think that's a good question. I think we're seeing a shift, but traditionally speaking, most data generated would serve the purpose of running a drug discovery program, right? These programs are really expensive, so we would try to sort of leverage the capacity that we have towards progressing a program rather than training a machine learning model. I think we're seeing a bit of a shift. I think more companies are seeing the value of generating data sets specifically for training machine learning models. There recently was a competition announced on Kaggle by Leash Bio. They have an example where I think that seems to be like very thoughtful of like, this is meant to be a chemistry or like a machine learning for chemistry data set. So yeah, I think that's the reason why we're not seeing a lot of it, but I do think we're seeing a shift. Thank you.
Thank you. A follow-up on that question, and the fact that of dataset releases and competition, is there any hope, in your opinion, to see like competitions where data is provided by a pharma company or biotech company, and then generative models are then tested, like molecules are synthesized in the lab and tested in the assays, or is that something that we can only dream of?
Yeah, good question. I don't think it's entirely unrealistic. It's definitely ambitious though. And I'm not sure like within what time span we'll see that work out. But like, I think one of the examples where they're at least trying to do something like this seems to be cache, which is another challenge similar to like CASP, but then for hit finding. And there they actually, I'm not sure if they do synthesis, but at least they let you like select molecules that you think are hits and then evaluate them. Does that answer your question?
Yeah, that answers a bit. Because I think like there's always going to be a limitation as long as there's a held out test set, right? There needs to be a lab experiment to really validate. The prediction, especially with generative AIs these days, like it's or even virtual screening, like we don't want just to do property prediction on a small set of molecules, but ideally either virtual screen or generate. Thank you. We'll go with the next question.
Thanks. That was a great talk and sounds really exciting. I guess I have two questions. So the first one is, do you plan to have support for data set and split versioning on this platform? Because I would say for the first one, people tend to disagree about the quality of assays and stuff like that. So different individual modules. And then for the split versioning, people also disagree on the best way to evaluate the models for particular tasks. And then the second question is, if Valence were to like no longer be interested in this area, do you think there's a way for the community to basically take everything that's been implemented and kind of mirror it so that it can be preserved, even if the industry interest is kind of diminished?
Yeah, no, so great question. Actually, I completely forgot to touch upon that. So this is lovely. In terms of the second question, it's all going to be open source and open science. So right, we use services for the web platform specifically that requires us to pay some fees. So it's not really easy to open source that. People like, we can open source it, but people won't be able to run it locally without the necessary API keys and whatnot. But the intention is really for this to be open source and open science. I can't stress it enough. I think it's really important that this is like community driven and not a single point of failure with one company that sort of like controls all of it. No, it really needs to be open source and open science. For your other question on versioning, yeah, that's a great question as well. So we don't have versioning as in you can upload one data set and there's multiple sub versions. However, what we do have is once you upload a data set, you can no longer change it. So there you can do some sort of like implicit versioning, but just uploading 3 times a data set. And there's no limit on the amount of data sets you can upload and just naming them V1, V2, V3. This is a very deliberate choice. I think we want to make sure that it's a source of truth. So if you try something again in like a year from now, it's the same data set. And the question was like, benchmarks are based on data sets, results are based on benchmarks in the way we've implemented it. So if you change the data set, what sort of happens in a cascade down that line? So we decided to make it just very clear, like once you upload it, you cannot change it anymore. Yeah, does that answer your question?
Yeah, awesome. Thank you.
Cool, thanks.
Hi, thanks again. Great talk. Synthetic chemists working with the group on some analytical methods. To touch on a point there you mentioned about synthetic accessibility and data sets, wondering if some of the data sets that are being introduced consider the impact of negative reaction data and that impact. I know with your background from computer science, that would be interesting. Wondering if you could just elaborate on the importance or role or absence of negative reaction data for these data sets.
Yeah, so I can answer that, I think, in a pretty straightforward way. So no, we don't have that right now. So there is a focus on like small molecule predictive modeling, and we're working with people to upload some of the data sets ourselves. However, I do want to invite you, like, if you think that's a data set that should be on there, even if you don't have time to do that yourself, point us towards the right resources. You can do this through GitHub or Twitter or whatever you prefer. And we're happy to like look into it and like see what how much of that we can do to kickstart the platform. Cool. Thanks.
Thanks so much again for the talk, and looking forward for people using Polaris. A big round of applause for Cass. So now we're on time. That's great. So you can refer to the schedule to see like where we are in terms of like poster session and lunchtime. We'll have now the poster session that will last roughly 1 hour. We have moved the poster around here so that there's some space. Again, to reiterate that we're presenting posters 1 and 2 for accepted papers, but also 3 and 4 for the spotlights. And I would like, yeah, so please enjoy the poster session. It will last for 1 hour before lunchtime.
Testing, testing. Hello, hello.
1, 2, hey, yeah.
Hey everybody, as most of you probably already saw, there are lunch boxes in the back corner. We'll keep the posters up throughout lunch, so feel free to keep chatting and discussing, but please go to the back corner and grab yourself a lunch box. We also should have enough for seconds, so if you're hungry and you want more, feel free to go ahead and grab a second lunch box too. We'll be starting again at 1:15.
Hello? Hello, everyone. So the conference will be starting back. The next speaker starts in 5 minutes. So please wrap up everything and yeah, see you soon.
Okay, mic 1. Yes, online 2. Okay, here we have mic 2. Mic 2 check. Et mic 3. Yeah. There. Check mic 2.
From here and how to use this microphone, or if you prefer,
Let's talk it out. I mean, it would look nicer. Yeah. Should I?
Yeah. Just give us 1 minute.
Yeah, no worries.
Amazing. We're still gonna share the screen on
But I'll share on the
Zoom and we'll
share that. Yeah, that's
you
better.
can just hide
Let's do
the.
that.
No, just leave it like this because we're
Yeah,
recording.
cool.
Just hit play as you did.
Yeah.
Just go down there and hide just go more and then hide floating meeting control. Yeah. And then if I plug this in, we'll be able to just
Well, I can just share my second screen. Let me share my
Yeah,
second
if
screen
you want,
because
you
then I can have then I can do
Yeah.
so sorry, let me connect this.
Yeah.
Give me a second. Wait 1 second. Okay.
We will be starting in 1 minute, so please take a seat.
Yeah, but this is the thing that you could find if you search around in recursions, like cool stuff. It's fun.
All right. I hope everyone had a great poster session. I saw so many people discussing and talking around brainstorming. So I love to see the life in the room here and everyone discussing about like the latest, coolest science. Now, before introducing the next speaker, I just want to remind a bit the schedule. We'll have 2 more speaking sessions of roughly 40 minutes each. So to leave maybe 5 minutes of question after the presentation, maybe we should leave the presentation for roughly 35 minutes. If you have a question, please line up to one of the 2 microphones. But we will leave the questions for after the presentation to avoid any interruption. Yeah, so I'd like to introduce you to Jason, who will talk about efficiently detecting interaction from high-dimensional observations of pairwise perturbations. So Jason is an incoming fellow at the University of Manchester, starting in October 2024. So maybe, are you taking students? Yes. Anyone looking for a PhD is your man. And he's also a research unit lead and staff research scientist at Valence Lab. Previously, he was a postdoc with Joshua Bianchio at University of Montreal. And before joining Mila, he was a PhD student at the University of British Columbia with Kevin Nathan Brown. He works on developing techniques for causal representation learning and causal inference. And a special thank you to Jason for filling up that spot last minute. It's but he's a great presenter, so I'm sure he has everything ready.
Great, thank you so much for having me. Yeah, so this is all pretty last minute. So you know, if you spot some typos on the slides, tell me and I'll be nice and embarrassed. Okay, so my talk today is going to be maybe a little different from what we've seen so far because it's thinking about more about the observations than what the perturbations are themselves. So I know MoML's primarily about thinking about molecules. And here we're thinking more about like, well, how do we measure the effects of molecules? And sort of what is the, specifically in these high dimensional observations. And the main motivation for this is we can collect, you know, these unstructured measurements of cells very cheaply, right? And we can collect these measurements tend to be like really information rich, and they're relatively easy to incorporate into these like highly automated pipelines. So I've spent the last year at Recursion, or at Valence, which is connected to Recursion, where they have these sort of amazing robotic labs for imaging cells at scale. And so that you can really collect a lot of data. But the data you get out is this sort of unstructured image data, which kind of presents challenges, and that's what we're going to be talking about today. First, kind of to motivate what I mean by information rich. I find this kind of fascinating that if you choose the right embedding of these measurements, you can get sort of surprising amounts of information about the underlying biology. So in particular, if you take kind of cosine similarities between embeddings with a really large model, and you look at the cosine similarities between sort of genes on the same pathway, you'll find that they tend to be highly cosine similar. And this is maybe obvious in retrospect, but I found this surprising when I first saw it. The idea is if you have sort of 2 genes that are coding for proteins that form a complex, I knock either one of them out, I've disabled that complex. And so I'm going to see similar morphological features in sort of image space. And so maybe it makes sense in retrospect that like they should be highly cosine similar, but I still find it surprising that we're sort of reasoning about things that we can't see visually, you know, in the pixels in the image, but we just sort of can reason about this from the sort of morphological changes to the cell. Okay, but all of this is operating at the level of single perturbations.
So cosine similarities will tell us kind of the effect of individual molecules or gene knockouts on the cell. But if we want to sort of start to think about combination therapies or extract any kind of causal understanding of what's going on within a cell, we need to find a way of getting past sort of single perturbations. And this is a problem because, you know, we can scale to single perturbations, but scaling to even pairwise perturbations is really, really hard, right? That just the combinatorics are against us. If you wanted to knock out all pairs of genes on the human genome, that would be on the order of 200 million experiments. And that's to say nothing about like kind of libraries of molecules where you might want to do pairwise molecules. And that's just way bigger than any experimental lab can do. And so this becomes an active learning problem, essentially. You want to choose pairs of perturbations that allow us to find kind of interesting drug combinations or interesting pairs of perturbations. But it's kind of an odd one because we're getting measurements in image space rather than measurements in the sort of traditional low dimensional space, right? And so kind of the more fundamental question in some sense is, if our image outcome is, if our experimental outcome is an image, how do we even define what it means to be interesting, right? So what are we searching for if our image outcome is an image? The basic mantra that we've taken in this work is that we should experiment where outcomes involve unpredictable interactions. We're going to spend most of the talk defining what we mean by that. And then we should just generalize to the rest, right? So machine learning tends to be really good at generalization. Let's kind of generalize where we can and experiment where things are unpredictable. And what I'm going to argue is that the surprising implication of this approach is that this can tend to lead us to experiment on things that are actually biologically interesting as well, right? And that's kind of the part that is more surprising, right? Okay. So how do we reason about, or how do we think about this problem? Well, the way I tend to approach this problem, coming from a causal inference background or causal representation learning background, is we think that the cells that we're imaging have some latent state. We're not going to specify what that is. We're just going to say there exists some latent state that describes the cell, which we use Z to denote. And then the images we observe are sort of pixels in an image that are sort of some rendering of that latent state, right? So we'll use the function G to describe the process of imaging the cell into pixels. And we can think of this as like a high-dimensional kind of readout of the underlying cell state. And then we're going to do perturbations on this on these cells, right? So we're going to perturb them by knocking genes out or applying different molecules, and that's going to push around this latent state by deactivating pathways or interacting with molecules and so forth. Okay. And the main assumption we're going to make is that the pixel level information is independent of these perturbations if we were to know this latent state, right? So kind of the latent state is capturing all the changes. We're not doing anything. We're not affecting pixels directly. Okay. And then, you know, the question we're trying to ask is sort of the most basic question you might want to ask is, do two perturbations have independent effects on the cell? Okay. Now this is a question that kind of you asked frequently. So in drug discovery, you'll ask things like, do you have synergistic or antagonistic relationships between molecules? And usually this is measured using something called a Bliss score, which I'll explain in a minute, which talks about sort of independence in terms of cell death. In biology, they do similar things where you're interested in whether pairwise knockouts of genes lead to so-called synthetic lethality, which is where cells die at a rate that is unexpected compared to what you would have expected from single perturbations on their own, right? So if I knock out gene 1, maybe the cell's fine. Knock out gene 2, the cell's fine. If I knock out both, the cell dies. Could be an example of synthetic lethality. And they measure this using independence tests on cell viability, right? Now, at its core, these two things are basically testing the same thing. Maybe it says something about people working on drug discovery and biology that one talks about cell death and the other one talks about cell viability. Optimist, pessimist. But really, it's the same independence test, right? You're measuring, if you're measuring the fraction of cells that survived, you're measuring this this kind of classic, something you would have seen in first year statistics, which is, can I move my mouse? Never mind. Which is just a product of two Bernoulli probabilities. If the two things are independent, you've got just a product of probabilities of Bernoulli random variables. And if not, so you're checking whether the fraction of cells that died in the double perturbation is equal to the product of the fraction of cells that survived in perturbation 1 and the fraction of cells that survived in perturbation 2. On the other hand, if you had chosen to write down the number of cells that died instead of the number of cells that survived, you're checking an or relationship because you could be either killed by perturbation 1 or killed by perturbation 2. And in which case, you're comparing in the double knockout perturbation, you measure some fraction of cells that died, and you're comparing that with the sum of the fraction of cells that died in perturbation 1, fraction of cells that died in perturbation 2, minus the products of probabilities to get rid of the interaction term. Okay, so this is very simple first year statistics stuff, but I guess the thing that's sort of interesting to think about here is when we're in control of this measurement process. So in this case, the state of the cells are the same. The things that are sitting on a petri dish are the same. And what we've chosen to write down changes the test we run, right? If we've chosen to write down the fraction of cells that survive, we run one test. If we've chosen to write down, fraction of cells that die, we run a different test. And the challenge here for us is that now we're going to work with pixel space, right? So we're not in control of that measurement process anymore. Okay, so we're just measuring pixels in an image. And sort of, can we still answer the same kinds of questions? Okay. The main idea that I'm going to show is that we can still answer these kinds of independence tests by looking at how changes in conditional densities are affected by perturbations, right? So why does this work? Well, I said before we had this latent distribution. So we have these we're talking about cell state in terms of this some distribution over latent variables. And if you remember the change of variable formula, that's good. If you don't, just as a reminder, we can express, we said that the pixels that we saw are some function g that is a transformation of this latent state. And the change of variable formula tells us that we can express this. We can express this conditional, this density over x as the density over the latents times this absolute value of the determinant of the Jacobian, which is a mouthful, but fortunately, we're going to get to kind of cancel that term out, so you can largely ignore it for our purposes. And so we can kind of talk about what we're going to be trying to do is talk about the changes in conditional density in terms of these changes in latent state rather than in terms of pixel space. Thank you. That's great. I can now point to things. Okay. So for simplicity, let's assume that each perturbation only affects a single latent variable. This is going to make it easier to explain. And furthermore, that this latent variable Zi is a leaf node, right? So if we were sort of describing the causal relationships between these nodes, we're going to assume that we're working with just one of the leaves. And then we can sort of write out this latent distribution as just a product over the distribution over any given latent variable given its parents. And when we do a perturbation, we only affect this leaf node's distribution, right? Because we've assumed that Zi is a leaf node, and we're perturbing only, and kind of by the causal factorization, we're only perturbing one of these terms in this joint distribution, right? And so so if I look at the density ratio of the perturbed cells to the control cells, it's going to have a nice simple form under these assumptions, right? So I can just compare the perturbed to the control from what we sort of saw on the previous slide with the change of variable formula. We have this is just literally writing out the change of variable formula, and all of this, all of this stuff on the right here is going to cancel, right? Because that's all the same. And so all you're left with the sort of change in distribution that you're inducing with a perturbation is just a function of of the sort of variable that you've perturbed, the latent variable that you've perturbed, and nothing else under these assumptions. Why is this useful? Well, it lets us test for independence of effects by testing whether the product of these density ratios equals the density ratio of the double perturbation. So sort of exactly analogous to what we had seen with the sort of Bernoullis, we can now do this in terms of density ratios. And so we can see this over here. So on the previous slide, we just argued that this density ratio for leaf nodes is going to be equal to that. Similarly, this density ratio for another leaf node would be equal to that. And if these two things are independent, then you're just going to have the same thing on the right-hand side, right? And so this is sort of like if you have independence, you have this nice relationship that is testable. Okay. Okay. So that gives us kind of a signal for when two perturbations are independent. We now want to turn this into something analogous to a test statistic. Because this is just something we can evaluate on an individual image, but it's not going to tell us what's happening at a distributional level. So we want to turn this into something like a test statistic. And full disclosure, we're sort of still actively working on this, and there are a number of design choices that you could make here. A simple thing you can do is just taking the expectation with respect to the control distribution. So this is just evaluate this expression on samples from the control distribution. And then if you just sort of write out that, and we do this in log space rather than the sort of original density ratio space, then what you essentially have is testing this relationship of KL divergences. Okay, so just kind of plugging in this expectation over density things, doing it in log space, you're essentially testing this relationship between KL divergences. And so this then becomes a test for independence, which we can then use downstream. How am I doing on time? All right, good. Okay, so that's one notion of independence. I'm going to tell you about a second notion of like independence, which I will be the first to disclose is a much weirder one to think about, but it has a nice interpretation in terms of composing embeddings, and so it's worth looking at. So rather than testing independent events, we can test disjoint events. These are sort of two different notions of non-dependence by testing whether the density composes additively. Okay, I'm not going to go through the details, but this is very analogous to what we saw on the previous slide. But whereas in the previous slide, we were talking about whether log densities composed additively, here now we're just talking about whether densities themselves compose additively. So sort of if you have this relationship between densities, then we can show that any embedding that you have of these cell images will compose additively in a way that you would like. So sort of in a way kind of analogous to kind of word vector addition that we've all seen in machine learning context, right? So if you've ever seen the sort of man is to woman, king is to queen kind of relationships that come up in word vectors, you can do the same thing sort of under this distributional assumption. You'll get the same thing with embedding. So the difference between the control embedding and perturbation of gene 1 and the control embedding and the perturbation of gene I and gene J, summing these up will give you the double knockout, right? This is a strange distributional assumption, and we still don't feel like we understand it well enough. But this fact that you can do this for a lot of pairs of perturbations shows up a lot, right? And there's a growing set of empirical evidence that we're able to compose embeddings of this form for a lot of perturbations, not all perturbations. Clearly, interesting interactions are the things where this fails is where things are interesting. But there's a sort of surprising empirical observation that this shows up a lot, and this gives sufficient conditions for this to happen, right? We can test whether this holds explicitly. Yeah. Oh, sorry. Okay. So this relationship that I've said holds for all embedding functions H, right? So it's not sort of particular to some particular choice of embedding function. This will just hold generally. But that includes sort of trivial cases like an embedding function just sets everything to 0. If if you want to instead turn this into a test for whether this distributional assumption holds, you want to search over all possible embedding functions and try and find the kind of the worst case one and see whether it still holds there. And that ends up being equivalent to what's called a maximum mean discrepancy test, which is just a test for independence of test for test whether two distributions are the same. And it's essentially these two distributions. That's equivalent to testing these things. Okay, so we can show this as well. Okay. So sorry, that was a lot of math, but we're now going to switch gears into results. So first, sort of just simple synthetic examples for whether these kinds of tests actually show what we would expect when we know the data generating process, and kind of indeed they do. The KL divergence thing is more sensitive than we would like, less sensitive than we would like. We're not seeing all the dependence showing up as strongly as we would like, and so that's why we're sort of actively working on different methods. Whereas the MMD thing is very clearly kind of detecting dependence where we expect dependence, but we're actively working on improving these methods. We then went into this on real data. We've done this for all pairs of 50 gene knockouts, as well as all pairs of 50 compounds. So you're sort of looking at compound combinations and then also gene compound pairs. We're going to look at genes in these slides, but we have results on the compounds and the gene compounds as well. And so this on the right here is MMD. So that's the thing I showed previously. And actually, interestingly, you get sort of a pretty similar pattern of interactions if you just look at the final hidden layer of a classifier. And there are reasons for that, but I won't go into that. Okay, so I'll show you sort of that on its own isn't that meaningful. It's just like a pairwise plot. But what's interesting is to then go and look at sort of what are the highly interacting pairs. How do they? Can we say anything about what seems to be interacting strongly? And this is a plot which I'll be the first to admit is not the most interpretable plot to read, but it's the thing that kind of got me interested in understanding what was going on under the hood when we first made this plot. So let me try to talk you through it. On the x-axis, we have the cosine similarity thing that we saw on the first couple of slides, right? So this is sort of how recursion finds interactions in the sense that we talked about before. If things are highly cosine similar, so like points out in this region, we'll say they're likely interacting because they have similar morphological effects, right? So they're probably on the same pathway. So that's the sort of cosine similarity metric. And then on the y-axis is this difference in embeddings, right? So I said things are going to compose when they're independent, and when they're not going to compose, right? And so anything on the x-axis is kind of cheaper to collect because it's just single perturbations. So you can compute these things from single perturbations, whereas on the y-axis these are double perturbations, so they're much more expensive. It's going to motivate the last part of the talk, but it's showing up. Different things are showing up under these metrics, right? So these are sort of outliers in terms of single perturbations. These are things we would have called interactions under the original test. These are outliers in terms of the norm difference of the embeddings. And so these are things we're going to call interactions under these new metrics. And then this is stuff that would show up in both metrics, right? So now evaluation is hard, but what's interesting is the things that show up as highly interacting are things like pairs of genes on the apoptosis pathway, on the proteasome, whereas things that are showing up on over here are things are like on the autophagy pathway. It's at least kind of consistent with what we know about biology that you should expect more redundancy on the apoptosis and proteasome pathways. These are things where you don't want kind of some malfunctioning protein to cause the cell to kill itself, which is the apoptosis pathway. So it's like these are inhibitors, and so and so if you're looking for kind of synthetic lethality kinds of relationships, finding sort of things finding these pairs of perturbations being strongly correlated with the apoptosis pathway makes sense. That said, I mean, evaluation of these things is extremely hard because we're going to detect interactions that go beyond what is known in biology because we're measuring interactions in kind of changes in morphology space, which is more general than just cell death, right? So you can have sort of changes in morphology that don't lead to cell death, and so many of the relationships we find will not be already known in biology. Cool. Okay, so this is all fun, but pairwise perturbations, as I said at the beginning, don't scale, right? And so you know, it's in some sense academic because we can do this for pairs of 50 perturbations. How do we do this for the whole genome and the like? So this takes us back into the world of active learning, and I'll note that sort of like active learning approaches are often derived from kind of an information theory perspective in terms of finding examples that maximize information gain under some notion of information gain, right? This perspective is really challenging to apply in these settings because when your experimental outcomes are images, kind of every observation is information rich. And so you're kind of comparing high entropy things to high entropy things. Good estimates of entropy are hard in themselves. And so like reliable comparisons between these high entropy things are very difficult. But we've just derived a bunch of test statistics that kind of tell us where things are going to interact, right? So we've done all this work to derive these test statistics that kind of give us an indication of whether two things are interacting. And maybe to kind of drive the point home, if you think about just like looking at images like this, it's not at all obvious whether two perturbations have interacted or not. If the cell dies, maybe, but in general, like I've seen one perturbation, it changes the morphology slightly. I see another perturbation, it changes the morphology slightly. Have these two things interacted? I don't know. I'm just seeing a third, I'm seeing a double knockout which has a small change in morphology. So even defining what it means to be an interaction is not obvious, but we've just done a whole bunch of work to define that, right? So and as a result, we've got like a set of test statistics that in some sense define a reward that we can use, that we can aim to maximize. We want to find places where things interact. And these will be the things that are unpredictable according to our test statistic, okay? And so what this does is it lets us reduce the problem of like active learning from images, which is a really hard thing to think about, to a problem of active matrix completion, where we're sort of sequentially selecting experiments to run in this sort of pairwise matrix, right? And we've got a score for every pair of experiments. And that's a much better, well-studied problem. Okay, so the approach then is we're going to learn a reward model that's going to predict this test statistic for every perturbation, every pair of perturbations, right? And so in this context, this is literally just matrix completion, right? So kind of inductively, let's assume we've selected some set of pairs of perturbations. Obviously, this works when we haven't done anything, but it's easier to think about once we've got some of this empty matrix filled in. And we can do matrix completion to kind of predict the rest, right? And then at every round, we're going to select the index that minimizes instantaneous regret per bit of information gain. That's a mouthful, but essentially this is a way of trading off exploration and exploitation. Okay, so we have our reward model that has some posterior over how likely we are to see an interaction. In particular, it's like a posterior over the test statistic for all pairs of perturbations, and you could sort of query it at every point. Some points will sort of be pretty certain that there's going to be an interaction. Some points will be very uncertain. And then what we want is points kind of like this, which are points where maybe we're going to see a strong interaction, maybe not. We're very uncertain. Querying that point will give us a large amount of information gain. And if it happens to be a good point, we'll select that point. Okay, so, that's and using this approach, we can very efficiently find interactions under our metric, right? So, so this is this on the left here. We're looking at the percentage of the highest the highest interacting pairs that we found over rounds of experimentation, right? So we sort of like take the top 5 percentile of test statistics, because we know all the test statistics in this experiment, and we see how quickly we find all of those in the top 5 percentile. And this information-directed search approach that we looked at on the previous slide tends to find these things very, very fast, faster than things like Thompson sampling and UCB, and certainly a lot faster than random search. In terms of known interactions, so we can also evaluate with respect to like how many of these interactions are actually known biological interactions. The differences are smaller, and this is kind of in part due to what I was saying before, is many of the interactions we're going to find are either false positives, like it's totally possible that we'll have false positives because there are some distributional assumptions that we've used. If those assumptions are wrong, those are going to lead to false positives. But alternatively, they're just novel biology that hasn't been discovered before, right? And so we really have to go and do some orthogonal assays to tell the difference between like this big difference versus this much smaller, more modest difference. Here's a fun animation of what the search procedure looks like. So this is sort of over experimentation. Random search, you can't see this that nicely, but random search is just obviously going to sort of randomly explore, whereas information-directed search is going to kind of like pick out these highly interactive genes and do more experimentation on those rather than on than doing it randomly. And so that's what explains the big gap between these two things. Okay, that's all I have time for. I just want to say thank you to all the various collaborators on this. Most of the theory around detecting interaction has been driven by David, who's in the audience here. The active learning parts was driven by Moksh, who's also in the audience somewhere, and Anikit. Sean and Ali played a big role in trying to stare at these results and trying to understand the biological significance of them. And then Burton has been with me on this project from the start and helped set up the experiments as well as been providing support throughout. And then finally, sorry, just some concluding thoughts. I think what's nice about this project is it kind of marries ideas from causal inference and causal representation learning, which allows us to kind of reason about where we can generalize and where we don't expect to generalize, and then design our experiments to go where we don't expect to generalize. I think it's interesting to think about how this generalizes to higher order interactions, so things like molecular fragments. We could probably think about in the same framework. And then as a sort of aside, this is more to my own community, the causal representation learning community. I think this work kind of presents a different perspective on causal representation learning. Much of the work in that community, including some of the work that I've done, focuses on explicit disentanglement. And that's a very challenging thing to do in practice, because you can't validate whether you've successfully disentangled something, right? So we see methods that seem to work nicely on synthetic problems, but then when you try and apply them in these complex high-dimensional settings, they can be very challenging to work with because you get an estimate, but you're not clear if it's right, and it's very difficult to test that. And I think what's nice about the testing perspective, so we're rather like kind of falsifying distributional assumptions rather than trying to disentangle, is it lets us, if we make incorrect distributional assumptions, it's just gonna lead us to false positives. But this is something we can then go and test with orthogonal assays. And it also just makes it far easier. We can actually validate our machine learning methods with actual explicit validation sets. This is very important when you're trying to fit these high-dimensional models, and it can be very difficult to do in standard disentanglement approaches. And so, yeah, so the thing, kind of this research program that I'm excited about is kind of thinking about like how observable quantities that we can estimate, like these conditional densities, can be validated and allow us to kind of extract the causal relationships that underlie these observations, these high-dimensional observations. Yep, that's it. Thank you.
All right, thanks a lot for the very interesting talk. Is there any question? Please come to the microphone.
Thank you for. Hello. Thank you for your talk.
could you go back to the one of the first slides when you introduced the diagram, the DAG structure?
Yeah.
Yeah. Here. So trying to understand why the arrows are not the other way around, at least between X and Z.
Between X and Z.
Yeah, because I mean, so there's two things. First, like Z is a latent state.
Yeah.
This is an observable.
Let me try and see whether so you know, we're thinking about the direction of information flow, the kind of causal direction of information flow. So, you know, the cell lives on a plate, and then we are imaging it, like there is sort of light that gets, you know, this function G is kind of measuring the process of like imaging that onto pixels. And so that's why the, you know, the causal direction is in that direction.
That makes sense. But okay, what I'm trying to understand, so you're assuming that the latent state partitions into a set of Z variables.
Yeah.
Like I'm wondering how reasonable it is to make this assumption in practice. We have a cell that, for example, or that kind of has a state where all these things are interacting with each other. Like I'm wondering if the partitioning assumption makes sense.
Good. Okay. So this is a great question. It's extremely subtle to think about these things. The way I like to think about latent variables in this context is as kind of morphological features of the cell, right? Things that you could point to and say, that cell is dying. So it's, you know, it's clearly a dying cell. So we have a sort of latent variable that describes that state of the cell. That cell is experiencing autophagy. You know, there's some morphological features associated with it. These things may be interacting, and these are kind of the output of pathways that exist underneath it, which we can't directly observe. Much of the theory in causal representation learning depends on an injective relationship between latent variables and observations. And so, you know, if you could in principle say, well, the latents are the genes, or the latents are the proteins in the cell, but that then you clearly don't have an injective relationship. You know, these we can't with a microscope see what proteins are active in the cell. We can only see their sort of morphological effects. But this is extremely subtle, and we could have a long conversation about it. So maybe.
One more question. In the later slides, you had an H function that you were optimizing over.
Yep.
You had a supremum over it.
Yep.
Not here.
I know this. Okay. So that's.
I was wondering what was the family of functions H that you were sort of optimizing over in practice? Is it like linear functions?
So it would be kernel. It's a kernel method. So it's, you know, so you're looking at like RBF kernel, you know, space of functions that are defined by RBF kernel, matern kernel, or the like. So it's the reproducing Hilbert space.
So you're optimizing credit in that case.
Yeah. So it's just a test, but you're basically looking for a test function. You're looking for worst case test function.
Does this guarantee that it's actually sufficient to have making up over the expectations in that?
So
then it applies for the density functions.
Yeah. So this will work if you've got if it's kind of a rich enough family of functions, basically. So you need a kernel that is recursive enough. So yes, and you know, if you're prepared to assume that.
Thank you. That's it.
No worries.
Hi, nice talk. I was wondering if your computational framework was able to differentiate between two types of genetic interactions. There are like positive genetic interactions, which are between synergistic genes, and there is the other type, which is between antagonistic genes. So I wonder.
Yeah, that's a great question. So sort of, can we? I'll just repeat the question because Mike's a bit soft. But can we tell the difference between a synergistic relationship and an antagonistic relationship, or the like? The short answer is I don't know how. The long answer is I think that it should be possible by combining what we observe from a single perturbation, these sort of cosine similarities, and these scores. So kind of combining these different scores. so single perturbations will say, if I've got sort of like two single perturbations that are very highly cosine similar, those are having like similar direction effect. But then if they're also having a very surprising interaction, that would then suggest that there's like an antagonistic relationship, because it's like you would expect these things to compose, but in fact, it's sort of they don't. That kind of thing seems plausible, but we haven't worked it out yet.
Another question. So,
Yep.
You touched upon redundancies, functional redundancy. It's very well known that duplicate genes, there are lots of them. They are enriched with synthetic lethality. So, I wonder how much proportion of.
Could you speak up a little bit? Sorry, I'm just.
How much proportion of synthetic lethal interactions that you found are between duplicate genes, like redundant genes?
That's a good question that I'm not sure about. So we didn't originally, when we chose the 50 genes that we knocked out, we chose them before we decided to look at synthetic lethality as being something of interest. And so there are a couple of synthetic lethal pairs in that set, and we do find that they tend to have large scores under these metrics, but we don't have like a set of genes that we know, like a large set of genes that we know are all synthetic lethal for the pairs. And so it's sort of hard to answer that with any kind of confidence.
I think great talk, especially the push for more testable implication of possible representation. I think it's a great direction. If you go to the slide with the active learning benchmark.
Yes.
This one. Comparing, for example, the random sampling, for example, the IDS. What kind of test function was that? Was that real, well inspired?
Sorry, this one? Yeah.
So what's on the left? What kind of test function is that?
Over there. When you say what kind? So this one was using the final hidden layers of the embedding function. So this is testing the non-interaction. So the disjoint setting. We're busy actually reproducing these figures for the MMD test and for the KL, but we haven't done those yet.
And on the right here, the confidence intervals of B5, because anyway.
Yeah, that's a good question. So like, there's a sort of big overlap in I think so. The right-hand side is this percentage of known interactions. Like I want to report it because I want to be upfront about it, but it's subtle because of this problem of like evaluation. Like really the thing to do is we see some interaction, we should then go and say do some orthogonal assays and see whether these interactions are real or not. Kind of evaluating us with respect to known interactions, you would hope we find known interactions, like that you would expect, but like we will find many other interactions which we say are interactions, and they are. And even if they are real interactions in a morphological space, they may well not be known in biology. And so, yeah, I would expect like false positives are going to make us look bad on that metric, and sorry, false negatives are going to make us bad. So they're like they're real relationships, but they're just not known to biology. Will look bad on that metric, and so it's hard to tell kind of how much of these are false positives versus just bad metrics.
Just looking at it. What color is UCB? Is it green? Is that worse than random?
No. So random was the that is weird. Sorry, that UCB was definitely better than random. So that seems. I think might just be rendering weirdly. Oh no. Okay, no, that is very strange. I was having a look at it. Yeah, I would expect UCB to be better. Cool. Thank you.
Do you have the question? Do you have a question or no? Okay, there's no more space. Okay, thanks a lot for the talk. If I may also ask a question. So you show that you can discover like when there's an interaction between the genes by looking at whether the sum of the vectors correspond like to the actual effect. But I'm wondering, like, especially when you consider that in many cases, these interactions are like, okay, well, the cell resisted to one intervention, but when adding a second intervention, then like the cell died, for example. Like, do you think that the effect that you would observe on all single genes would be visible enough from like the batch effect, for example? Like, I think like if you were to test to take the same cell and like clone the I don't know, like clone the universe and reproduce the same experiment, it would make a lot of sense. But considering that there's so many noise in the experimental setting, are you actually like, how confident are you that you can really take these vectors and sum them up or subtract them to retrieve interactions?
Yeah, that's a great question. So, I mean, I think to deal with batch effects, we end up having to do a lot of averaging, and that's true of both the cosine similarity relationships and the things. So, but if you look at, I don't know, this plot isn't great, but if you just stare at this plot, you can see that we get sort of close to zero predictions for a lot of things. For a significant part of this matrix ends up being close to zero. To do that, you have to do averaging over a number of different guides and a number of different replicates in order to sort of average over many of the batch effects, and then you start to get more reliable signal. But even so, like, you can certainly have the case where, you know, a perturbation that has a very minimal effect on the cell could then be sort of end up having sort of pointing in random directions, and then will not be a reliable signal. I think the right way to deal with that is just having a good kind of pre-processing step that is checking whether you're getting reliable perturbation consistency, and then only make these conclusions on things that are sort of reliably perturbing things.
Thanks so much for the great talk, and another round of applause for Jason. And congrats again on the professorship position.
Thank you. Thanks.
So now let's welcome our next speaker, Raquel Rodriguez Perez. All right, so Dr. Raquel Rodriguez Perez will be presenting the talk, Leveraging Molecular ML and Property Prediction in Drug Design. Dr. Raquel Rodriguez Perez is an Associate Director and the Senior Principal Data Scientist at Novartis Biomedical Research. In her current role, she integrates AI technologies into decision-making in drug discovery's project. She enables AI-driven drug design by leveraging techniques such as molecular property prediction, generative chemistry, uncertainty estimation, and explainable AI. Raquel obtained her bachelor and master degrees in biomedical engineering from the University of Barcelona and her PhD in computational life science at the University of Bonn. She was also a Marie Curie Fellow and worked at the computational chemistry data science team at Boehringer Ingelheim in Germany. Her research has focused on predictive modeling and pattern recognition for different applications in chemistry and life sciences. So I'm really looking towards that talk, because that talk, I think, will open our eye into how to best use these kind of machine learning techniques that everyone is presenting, but in real world industrial scenarios. Thank you, Raquel.
Thank you. Thanks a lot to the organizers for the invitation, for the kind introduction as well. Looking forward to explain you a bit how we apply machine learning models in pharmaceutical research. In Novartis, we have been leveraging molecular machine learning for many years. I've been 4 years in Novartis, and I've seen a lot of applications in real-world drug discovery, and I've seen a lot of impact of machine learning, a lot of opportunities to accelerate and improve drug design. Today, I would like to give you an overview, but also highlighting the opportunities and challenges. I will highlight some open questions and I think relevant research areas from academia also that we can use later on in industry to improve our pipelines. So first of all, let me give you an introduction on machine learning in drug design, how we are trying to incorporate machine learning in our drug discovery pipelines. So first of all, I would like to highlight that the decision-making in drug discovery requires the combination of data, which can come from in silico, in vitro, or in vivo experiments, And you will have different amounts of data with different types of uncertainty at different stages of drug discovery. So you need to take decisions based on this. And machine learning, it's one of the ways of obtaining some of this information. And when we have some specific targets and we are designing molecules for these targets, we will need to score them according to some properties that we need to optimize. These designs can come from a human or from AI, but they will need to be scored in the same way. And actually, this property optimization and the prediction models that allow us to have an idea of the properties of the compounds that we are designing will be the focus of this talk. Afterwards, you can also make predictions of the synthesability, and you can measure some compounds either to try to find your development candidate or to improve your models in the so-called active learning loops. I'm sure you are all familiar with molecular machine learning for property prediction, but just to be all in the same page, these are the type of models that I will be talking about today. These are models that relate the structure of the compound to the property of interest. So they are called quantitative if they are regression, or qualitative if they are classification, structure property relationships. And for that, we will first need to represent the molecule numerically. For that, you can use more traditional methods such as fingerprints, where you encode atom environments, and you can also encode the presence or the absence of given chemical substructures numerically. And then you use this as input for a machine learning model. There are other types of representations, two-dimensional, three-dimensional descriptors, and the most recent learned representation that can come from embeddings of a deep neural network that has been trained on a large scale. For instance, one of the models that we are using a lot right now, it's the Melody. I don't know if you will be familiar with it, but this comes from a federated learning consortium of 10 pharma companies, where we trained models on a large scale, and we are able to use these learned embeddings from the Melody consortium to build our property prediction models. And actually, this will be one of the models that I will present today. Regardless of the type of molecular representation, the machine learning algorithm will relate the chemical patterns and the molecular representation to the property of interest. And it's really important to validate that the predictions that you are making are correct, and it's not only identifying correlations that are not able to predict for new series or new compounds. And now that we know which type of models I will be talking about, I would like to give you an example. This example is metabolic clearance. This is one of the main absorption, distribution, metabolism, and excretion properties, so one of a key ADME assay, in this case for metabolism. So we have some in vitro systems to assess the hepatic elimination of the drug. This is quite important because if we only focus on activity and we don't consider these ADME aspects early on, then we will have like a lot of chances of late stage compound attrition. So this is one of the models that I developed during my first years at Novartis. And I think the most interesting part is that we have a multi-species model because we had assays with similar protocols that were targeting liver microsomes from different species. In the table, you can observe that we had a lot of data for rat, but we had much less data for species like dog and monkey. So when we were building single task models only based on this assay data, we're having worse performance. And here you can see the results in the different panels. You have the different species, and we are reporting the geometric mean fault error, which is the relationship between the observed and the predicted. And we have compared different algorithms. We typically benchmark different state-of-the-art methods, in this case, XGBoost, a graph neural network on a single task, but we also built the multitask graph neural network and the multitask graph neural network ensemble. And especially for the species with less data points, we observed a benefit of multitask learning. I would like to highlight that the largest improvement was based on this multitask learning. So sometimes it's how you use your data and not the algorithm itself, because the representation either was the XGBoost with Morgan fingerprints or a single task graph neural network was not having a lot of difference in performance. And it was the leveraging of the different multi-species data that led us to an improvement. But this is only one property. We have a lot of properties to optimize. And for each drug discovery project, we will have a different target property profile. There will be a lot of commonalities, but also a lot of things that are specific for a project. For instance, if we are working on a project for a neurological disorder, we might want the drug to penetrate into the brain. So then our target property profile will be different. Maybe we have observed some liabilities for liver toxicity, and we also want to include this in our target property profile. So we have to define this together with the team, collect the required data, and build the required models that allow us to make good estimations of the properties. And for that, some things will be project specific, but the goal is to bring the ADME and PK properties to the target property profile, and if we are able to do it across all the projects. So now I would like to focus on some challenges and recent investigations that we did. And one of the aspects is this concept of the project specific versus something that is applicable across projects. And this led us to benchmark different training set compositions. If we have large amounts of historical data in pharmaceutical industry, shall we use all of our data for model building? Or shall we focus on the specific parts of the chemical space that we are exploring right now? Sometimes the protocols of the assays change over time. So maybe if we are merging all the data together, we are incorporating noise, biases, we are modeling structure property relationships of parts of the chemical space that we are not interested anymore in. And I think everybody had a lot of perceived advantages and disadvantages on this topic. This was quite a hot topic at Novartis because medicinal chemists often were requesting local models for their project or their series because they had the intuition that a global machine learning model cannot capture the subtle differences of the structured property relationships. On the other hand, if you ask to a data scientist or a machine learning researcher, probably they will tell you the more data, the better. So what shall we do here? So what we did was a systematic comparison, and we started building local models for the different projects and compared to a global model for ADME property prediction. And actually the comparison was quite extensive because we did it across 112 projects, 10 ADME essays, and approximately 330,000 compounds at Novartis. And I think the results are quite surprising because in the x-axis you have the 10 ADME essays, and in the y-axis you have the relative performance. And you can observe that most of the distributions of the relative performance are on the positive side with all the dots in blue. Each dot corresponds to a project. And this is indicating higher performance for the global models. So there was a clear winner. The global models, based on heterogeneous data from different parts of the chemical space, different therapeutic areas, targets, were clearly outperforming local models for specific projects. And what it was also quite interesting is that only in 7% of the cases, the local models represented an improvement of 20%. So now every time that someone requests a local model, I send the link to the paper, which has saved a lot of time, but it has also given us some time to explore whether we can do better. And actually for that, we started exploring transfer learning techniques. Because at the end, we are having a changing distribution of our data over time. We are moving towards other parts of the chemical space, and sometimes even new modalities. So I would like to show you some of the results that we got with these investigations. Perhaps you have heard about transfer learning, and in the field of cheminformatics, normally it is applied for new tasks. So basically you have a matrix where you have in your rows the compounds, in the different columns your normally activity assays for different targets, maybe for this different assays for the same target. And then you have a new assay of interest or a new target of interest. You don't have a lot of data, and then you start applying few-shot learning, transfer learning. And I think this has been quite explored in this field. But what has not been so much under investigation has been, what if we include new rows that are from another distribution? Can we adapt our models for the same tasks, but doing something differently, trying to adapt the domain of our model? And this is what we have done for ADME models. We have set our global model with our global data across all the projects and also the historical data that we have in the database. And then we also took some project-specific data, which sometimes does not even have any data in the training set. Sometimes it has solely a small fraction, and then try to apply some domain adaptation techniques. And here I will show you some examples for one property and a couple of techniques, but in the publication you can see an extensive comparison with many more methods and essays. Here we come back to the metabolic clearance model that I presented earlier. If you remember, it was a graph neural network. It was coupled with a feed-forward neural network. So we have this graph neural network for representation learning, and then the feed-forward neural network for the metabolic clearance prediction. And the two transfer learning techniques that we tried, or the ones that I present here, are the weights initialization. So we consider the global model weights as initialization, and then with the project-specific data, we fine-tune the complete network, and the results are shown in orange. And the other strategy would be freezing the message-passing neural network, so the part of the graph neural network, and then we fine-tune the rest. Again, for comparison, we also report the performance of the local models, and this is relative performance compared to the global. So again, we observe that the local models are typically performing worse, but actually this transfer learning with the additional project data, it's allowing us to have lower errors. So this is also an interesting strategy in case a project is facing difficulties with our ADME prediction models, because we can further refine, and we also were able to see that there was no catastrophic forgetting in the performance of these models. And what about new modalities? We have focused a lot on small molecules, on the space of the rule of 5, but now we have a lot of new modalities that are also in our pipeline, and one of them are the targeted protein degraders. We have two main submodalities that are highlighted here, the glues and the heterobifunctionals. The heterobifunctionals have the linker and have larger molecular weight. So we were expecting that maybe they are outside the applicability domain of our models, and all the TPD programs and Novartis were not using our models because we thought they were not applicable. And actually at some point we thought, let's just evaluate, and if they are not good, maybe we can apply the transfer learning as well. And these are the results that I'm reporting here. In green, you can see the results, the mean absolute error for the different essays in the x-axis. And this will be across all modalities. In orange, you have it for bifunctionals, and in blue, you have it for glues. Interestingly, we observed that the performance for glues were consistently quite low, and in many essays, the models were performing very well on the glues. For the heterobifunctionals, we also observed quite a good performance for many of the properties. And if we look at the 0.3, which was actually mentioned in Cass talk earlier, and it's normally the number of the experimental variability, so normally it's twofold in general. So if we look at this threshold, we observe that only in some cases model refinements might be needed. And in these cases, we also tested the transfer learning strategy with the fine-tuning. In this case, we tested two strategies. Both are fine-tuning on the complete graph neural network and feed-forward neural network. However, in one of the cases, we did it with the newest data. And in the second case, we did it with all heterobifunctional data. So we also exposed the model to some of the data points that were already in the training set, if they were from heterobifunctionals. And what you can see in the results, in the x-axis, you have the permeability, metabolic clearance for rat, for human, CYP inhibition, reversible, time-dependent inhibition, and reversible. So for all these properties, what we observed is that the performance on the global models, which is in green, was worse because this is the mean absolute errors. So the errors were higher consistently than any of the fine-tuning strategies. And depending on the property, one of the strategies of fine-tuning was better than the other, but there was not a real consistency across all the properties. So the take-home message from here is that we can adapt global models for specific projects, but we can also do it for new modalities. And actually, the QSPR models based on machine learning are applicable to TPDs. which is something that the community did not know so far. And another interesting aspect is that we are trying to work on ensuring that this kind of investigations can also be done in the public domain and that there are high quality data sets. And one of the strategies that we started publishing are the surrogate data sets. So we cannot publish structures from our internal database due to intellectual property. However, what we could do is to use our high quality models that we have developed in-house to annotate the structures in the public domain. And we have done this already once. This is published. We did for a CP inhibition model that we have internally, and it's published now in Chemical Research in Toxicology. We took more than 16,000 chemical structures from Chembel and PubChem, and we annotated them with CP inhibition predictions from our in-house model. What is interesting is that when we developed a model based on this surrogate data set, the performance was quite comparable to our internal model. So this means that we can explore additional ways of model building in the public domain and have these open source tools for further research. And related to TPDs, we are also attempting at publishing a quite large data sets with more than 270,000 chemical structures from the public domain, including products. And we have annotated them with 25 properties. So this will allow us also to explore machine learning models for target protein degraders. And now I'm going to discuss the last challenge, which is one of my favorite ones, which is the uncertainty estimation. I think this is quite important because we often talk about uncertainty in terms of model predictions, but we also have to assess the uncertainty in our data. And something that we should do, and we normally do at Novartis, is to compare the performance of our models with the experimental variability of the underlying data, because this is the upper performance bound that you can get. But now let's focus first on the quantification of uncertainty in ML models. So normally the property models are evaluated on a test set, which can be challenging, which can be with a temporal evaluation. You will get some performance metrics, but then you will get a new data point. For this new data point, can you trust the model? And there has been a lot of research, and actually the concept of applicability domain is quite old in cheminformatics. A lot of people use the distance to a training set. This is quite a good proxy. Normally, if you have the chemical space, your training set is covering part of this chemical space and you have a new compound. If it's close to your training set, probably the model does well. If it's far away, probably the model has more error. But what is the problem? That sometimes there are discontinuities in the space. You might have a compound very close to a training set, but maybe it's an activity cliff, property cliff, you cannot predict correctly. And sometimes you also have compounds that are far from the training set, but actually the model can extrapolate. And these are the best cases for using the model. For instance, in generative chemistry workflows, if you can generate new molecules and these are different from your scaffold or different from your decorations and are predicted well, these are the most interesting cases. We want to capture them. So actually, these database uncertainty quantification metrics are not perfect, are giving us some information, but are just a proxy. Now we also have the model-based UQ metrics. You can have the variance from an ensemble, you can have the mean variance estimation, you can do some bootstrapping and get a variance as well. There are many options, but if you look at the performance of these uncertainty estimates, sometimes are also not giving you a lot of information about how the model is doing in real life. So for instance, for the coming back to the metabolic clearance model, we had an ensemble, if you remember. So we have also the ensemble variants available. And we used a calibration set. In the plot, you can see how the fault error was changing if we were adding compounds with increasing uncertainty or decreasing uncertainty. So the first one, you see how the error is going down if you start adding compounds that have a higher variance, and then you start adding compounds with lower and lower variance. So basically, yeah, it's encoding some information. Of course, if you focus only on the high variance compounds, you will have a higher error. And if you focus only on the low variance compounds, you have a lower error. We tested whether this was also the same for a validation set prospectively. And yes, we were able to have better estimations in the low uncertainty compounds. So if you check the table, you see R squares close to 0.6 for the low uncertainty, and for the high uncertainty, you have 0.4, 0.47. So yes, there is some signal, but this estimation is not perfect. And probably depending on the data set, the preferred uncertainty estimate will also change. Here, I also have a comparison to experimental variability. I think it's quite important to observe that when we have a low uncertainty estimation, we are actually quite close to the experimental variability, or at least the Q3 of the distribution of experimental variability. So again, when building the models, it's important to have in consideration the uncertainty in your data and the uncertainty in your model. But since the uncertainty estimation is quite a difficult topic, we have created a framework for uncertainty quantification benchmark. We are going to release this Python library that unifies the benchmarking of UQ strategies. So basically, when we went through the literature, there were many strategies for quantifying uncertainty, but also many strategies to evaluate whether the uncertainty quantification was good or not. And I think the evaluation will depend a lot on how you are going to use the model. Because if you want to use your uncertainty estimation to flag some compounds and put them out of distribution, then maybe you should focus on one metric to evaluate the uncertainty. But if you are going to do an active learning loop, and you want to create your acquisition function, you maybe need well-calibrated prediction intervals. So all these aspects are considered in the design of this library. You will have a comprehensive evaluation settings that are going to capture the superiority of uncertainty estimation metrics depending on your application scenario, and you will get some recommendations on which metric to use. So just to give you a quick overview, the only user input will be some information regarding the model, such as the predictions, or for instance, if you have any ensemble variance, or maybe the posterior of the Bayesian model. And you can also report some molecular representations. These molecular representations can be the ones from the model, or you can also decide to bring a CDDD representation or molar. Like you can use whatever you want for representation. And then the library will calculate some uncertainty metrics. Some of them will be distance to a training set, kernel density estimations, and some of them will try to combine these two aspects of the database and model-based uncertainty quantification metrics. So we came up with some scores that are trying to bring these two aspects together, and we are currently benchmarking them using the library. Moreover, we have a special type of uncertainty quantification metrics that are the error models. So basically are machine learning models that predict the error of the property model. And these ones in our hands were also working quite well. But so far in our implementation, you need to take aside part of the data for the error model building, so you lose a bit of data for your property prediction model. And finally, as I mentioned, we have a quite complete evaluation module so that you have a nice benchmark of the uncertainty quantification metrics. And I would like to finalize the talk by moving towards in vivo PK predictions. Most of the models that I mentioned, or all of them, have been about in vitro ATME properties, but actually the in vitro ATME are also a proxy of in vivo PK in preclinical species that then will also allow us to predict better in vivo PK in human. The data that we have of in vivo PK in human is quite scarce and also very biased, because you can find some data in publication, but they are from commercial drugs and the chemical space that we are not currently exploring. So what we decided is to focus on predicting PK in preclinical species, because this data still covers the chemical space that we are currently investigating. And we have just put it on our ChemArchive a new methodology that we designed. This is to predict the concentration time profiles of the PK studies. So far, most of the investigations have focused on predicting clearance, predicting area under the curve, predicting derived parameters from the concentration time profiles. To the best of my knowledge, there was only one publication from AstraZeneca where they predicted a concentration at different time points, but these time points were specific. And what we came up with is a different strategy where we predict the constants of a PK compartmental model. So the compartmental model, it's quite used when you have the experimental data and then you want to fit it to do further simulations. Because once you have this compartmental model, you can simulate, for instance, multiple dosing. So in this compartmental model that you see in the slide, you have the central compartment, which is the blood, one first peripheral compartment that it's illustrating the highly perfused tissues, and then another one that it's illustrating the barely perfused tissues. And you have some constants that define ODE system. What we did is to predict the constants of this ODE system and then obtain the concentration time profile. The loss was calculated between this concentration time profile and the experimental one, and then we were back propagating. So at the end, you can obtain the concentration time profile, the compartmental model that allows you to do further simulations, and you can also derive the PK parameters. So far, we have evaluated this model for rat PK studies, and you see some examples of our predictions after IV administration and oral administration. Larger errors were observed when we were doing time split compared to random split as expected, but actually the results were also successful for after IV administration in a temporal validation. Oral was more complicated, but you can observe in the table that the experimental replicates were also illustrating the high variability of these studies. Another evaluation was done after deriving from a non-compartmental analysis, deriving the PK parameters from the predicted curves and comparing the PK parameters to the ones obtained from the original experimental curves. So we have here results for area under the curve after oral and IV administration, maximum concentration after oral administration, and so on. You can get a lot more results and insights if you have a look at the publication. But what I wanted to highlight here is that these results were close to experimental variability, and also that the DeepCT method that I'm presenting showed similar or superior performance than when we were training the model to directly predict the PK parameters. And finally, as I mentioned, we can simulate multiple dosing because we have access to the compartmental model, and we also did some tests with commercial drugs to see how the simulations were looking like. So to finalize, I would like to give you some take-home messages. First of all, that machine learning models only based on structure are quite powerful tools for prioritizing compounds before synthesis, and also to drive the generation of new compounds, either informing medicinal chemists how their designs are looking like in terms of target property profile, or informing the AI system on what to generate next. We have presented a new methodology to predict the PK in vivo, specifically to predict the concentration time profiles. And these in vivo PK predictions are not only helping to prioritize compounds before synthesis, but also to prioritize animal studies. And I've also talked a lot about global models and transfer learning. The global models have shown generalizability across projects, even when the projects do not have a lot of data in the training set. If you apply transfer learning, you can get improved performance. And these models have also been applied to target protein degraders and have shown successful performance for ADME predictions. And finally, what is very important in drug design is to combine the experimental data and also the predictions, considering their uncertainty. You have different data available at different time points and different stages of the drug discovery process. And if you integrate properly the uncertainty and the variability that these have, you can have better decisions. And to facilitate unified evaluations of these uncertainty estimates, we will open source a Python library that is called Unique. I hope I gave you an overview on how we are using molecular ML methods in Novartis, highlight some of the opportunities and challenges, and thanks a lot to all my colleagues and all of you for your attention.
Thanks so much for the great talk. A lot of the work that was done here also resonates with some of the work that I've done during the past year. So I found it very interesting, and we kind of also arrived to the same conclusion that retraining on many tasks, even if like they don't necessarily correlate, even if they come from different protocols, it's really helpful for the downstream model. One thing that I found particularly interesting is the uncertainty estimation that you work with, and how you combine uncertainty, like distance of the data and uncertainty of the model. And I'm wondering, like, where do you see that uncertainty would be more helpful? Is it, at least at Novartis, is it like to do some active learning to reduce the uncertainty, or is it more to discard whatever the model is uncertain? Rather than like maximizing knowledge?
I think active learning is definitely quite important, but for most of the models I'm working with, we already have a lot of data. And even though active learning loops are useful, I think what it's more critical for me is to integrate them in decision making, to make sure that when decisions are taken with the models, it's because the model is quite sure about it. Also, when we are integrating models with different applicability domains in a multi-parameter optimization in genetic chemistry, we need to start thinking more systematically on incorporating uncertainty, because if not, the algorithm is moving on directions that we are not expecting.
Okay, thank you. If there are any questions, please come to either one of the two microphones.
Great talk. I just had a question. So I thought the work that you had about the global property prediction, also like training on different tasks, was helpful. The fact that that's helpful is interesting. I'm just wondering if you see like a lot of benefit from doing sort of more self-supervised or unsupervised training just on like pure sort of structured data, so data that doesn't require any assays.
Normally, we always do supervised learning, trying to learn from the essays that we have carried out in the past and trying to predict. On this sense, it has been more on representation learning, I think, what it has been studied by other teams at Novartis, but I have not been involved in that. When I use unsupervised learning, is more on exploratory data analysis, but not on predictive modeling.
Thank you.
Hi, great talk. You mentioned Project Melody and federated learning. I'm just wondering if you've developed ways to estimate data uncertainty when using federated.
Sorry, in case I didn't understand properly. So you are asking if we considered uncertainty when using the Melody embeddings or the Melody models? Yes. Okay. Okay. No, so far, actually, I have not used any uncertainty estimation from the Melody models. Now that we have established unique, we can run unique without having access to the model. So only with the predictions from Melody, we could run unique on it. And this is something that is actually in our plans, but we have not done so far because we're working on the library. And actually, I mentioned that I said during the talk that I will mention work with Melody, and the work that we did with the Melody embeddings was actually developing this. So I think it's quite interesting to highlight now that you asked, because the representation that we use for the chemical structure was actually the Melody embeddings. So from Melody embeddings from a federated learning consortium of 10 pharma companies, then we were able to predict concentration time profiles of in vivo PK. So. But we have not propagated any uncertainty here.
Awesome. And unique, you're gonna open source.
Yeah.
Okay, Awesome, thank you.
Thanks.
Hi, thanks for your talk. I'm just wondering if for those global models, did you investigate different split methods, or do you just, because you have so much data, you can do random and it's gonna have the same impact, or do you need to cluster and ensure that you're picking up the most diverse set, for instance?
Sometimes for some benchmarking, we compare different splits. Like for instance, in the DeepCT here, you see that we also tested the random one. However, normally our gold standard is the temporal split. So we have the temporal information. We know when the compound was registered for the first time, and we also know when the compound was measured in each essay. So we normally do this time split to always ensure that we are evaluating the models in the same scenario in which they will be used. Yeah.
Thanks again for the great presentation.
Thank you.
So now we'll be heading for the last poster session, where we will present posters 3 and 4, and again, all the spotlights poster that stay for both sessions. We're running a bit late, so for the next talk, we'll be 10 minutes after the schedule at 3:45 for AlphaFold 3. So enjoy the poster session. Hello, everyone. So we will be back at the for the presentations in a bit more than 5 minutes. So if you can wrap up a bit everything, thank you.
Yeah, mics are aligned. Mic there, of course, yeah.
Check podium. Hello, est-ce qu'on m'entend bien dans la salle? Yeah. Et check mic 3. Awesome.
Hello, we'll be starting in 2 minutes. Please take a seat. For your last name, you pronounce Ja or Ya? I do Ya. Yeah. Okay. Okay. Perfect. All right. Please take a seat. We're getting started. So for the last presentation of the day, before we jump into it, I want to say, like, please don't run away when the presentation is over. We want to take a big group picture. So let's remember to do that. Now for the last presentation, last but not least, the presentation is towards rational drug design with AlphaFold 3. And the presentation is by Max Jaderberg. So Max is the chief AI officer at Isomorphic Lab, an alphabet company where he reimagined the entire drug discovery process from first principle with AI first approach. Previously, he was a research scientist at DeepMind and led the open-ended learning research team. Prior to that, he co-founded Vision Factory, which was acquired by Google in 2014, and completed his PhD in visual geometry group at University of Oxford with Professor Andrew Zisserman and Professor Andrea Vedaldi. His main research interest is in AI and deep learning for like drug discovery at the moment, and he will be presenting AlphaFold 3. Thank you.
Great, thank you so much. It's such a pleasure to be here. You know, I've been coming to this conference for the last few years now, I think since the beginning. And it's amazing to see how this forum has been evolving over the years. All of the incredible work that's being presented out there, very, very inspiring to see indeed. So I'm going to be talking on behalf of, you know, some amazing teams at Isomorphic Labs and Google DeepMind behind a lot of this work. And, you know, I've been at Isomorphic Labs since the beginning of the company. Started about 2.5 years ago, spun out of DeepMind. And really there, you know, our core mission is to think about how can we get therapeutics into the hands of patients at a scale and efficiency that, you know, you can't even think about today because of the many bottlenecks in drug discovery. And at the core of that, you know, we're trying to answer this question of, you know, how can we use artificial intelligence to redefine drug design? And, you know, I really use, you know, the word redefine in the strongest sense here. You know, I think there's a lot of opportunity to go into the workflows of the, you know, current medicinal chemists and biologists and the drug designers, go into those workflows and you know, create models that plug in and improve certain parts of that. But at some point, there's, you know, there's a bigger ticket item here, which is if our models, you know, and as our models get more and more accurate and start to be able to replace certain parts of the experimental pipeline, we really need to go into these workflows and completely shake them up and completely change the processes that go on behind drug design. So you know, a lot of us are from a tech background. I'm in particular, we all love this exponential growth in tech that we see, you know, particularly in compute power. You know, we just get more and more compute over time. And we know now that if we're able to, you know, create the right machine learning modeling paradigms, you know, I think of things like large language models of image generation, we're able to actually ride that exponential curve of compute really, really successfully. Now, on the other side of things, you know, I'm in drug discovery, where actually productivity has been on an exponential decline, right? There's no shortage of, you know, disease burden in society and our world, but the actual, you know, number of drugs being produced per $1 billion spent has been exponentially decreasing, right? So it's taking more and more money to develop new therapeutics. So, you know, there's maybe a big opportunity to bring these two worlds, exponential increase, exponential decline, together and see what happens. And I think that's, you know, really, you know, a huge opportunity out in front of us. So when I think about how we're trying to do this at Isomorphic Labs, you know, there's going to be a lot of different bottlenecks in the drug design process. And, you know, here you can see some of the different stages in drug design. You know, starting on the left, you've got things like target discovery and target validation. So that's thinking about, okay, you know, we want to target a particular disease, but what's actually, you know, the protein target that we want to modulate here that we think is going to affect the disease state, you know, of cells, of people ultimately. Then there's this middle section of chemistry. You know, once we have this target protein or system that we want to modulate, how can we produce chemical matter, you know, molecules that will go in and actually, you know, bind to these systems and change the way that they function. And then not just change the way that they function, but actually we want these things to be good drugs. So we have to do what's called lead optimization to make them better and better molecules that can actually put into people, maybe even take as a pill. It'll get to the right place of the body. It won't have any toxic side effects, and will be broken down in the right way. And then, of course, there's the whole business of then you've got to take these molecules into the clinic and see efficacy in real people. And that's a massive bottleneck. So there are these 4 bottlenecks we think about in general. The first is on understanding disease biology. What are the targets we should be going after and that are really going to change, you know, the function of disease? Then there's bottleneck 2, which is all about molecular design. Okay, we have a target. How can we design a molecule to go in and modulate these targets? Then there's the third bottleneck of actually assessing compound quality and toxicity. So as we put these molecules into people, you know, do they actually behave as we expect, but will they also interact with other parts of the body, other cellular systems, and have really adverse side effects that we don't want? And then there's another bottleneck, which is bottleneck 4, which is about, and I think this is more of an industry-wide bottleneck, which is that, you know, for some targets, you can spend 10 years trying to crack, you know, molecule design to get something that you might go into the clinic with. But at the end of that, do you get any transferable lessons? Do you build any transferable pieces? That means that the next target is going to be quicker and easier to work on. And I think it's this ability to scale drug design across many different targets that we really need to go after fundamentally. So we're doing a lot of research at ISO on all of these, you know, targeting all of these different areas. We've been focused a lot in bottleneck 2 on the chemistry side of things, but really, yeah, across this space. And today I'm going to talk about, you know, one particular area of research that we've been doing, which is for this new model AlphaFold 3, which is really targeting bottleneck 1 and 2. You know, this is the evolution of the AlphaFold lineage, looking at structure modeling. And this model, you know, first for bottleneck 1 allows us to understand much more about how, you know, the proteins and molecules implicated in disease interact within the cell. And then this crucially gives us clues, gives our medicinal chemists clues into how, you know, these molecular machines function, and therefore how we can go and modulate them. And then the second is that it allows medicinal chemists to actually design and test their designs, you know, potentially purely in silico, so they can see how changes in, you know, atoms in the designs of these molecules actually translate into the 3D structure that's forming with these targets. And ultimately, we believe that if we're able to do, you know, understand this biology better, if we're able to design better compounds, this will translate into higher success rate into the clinic and faster, more scalable drug design, which is ultimately better for patients. So structure prediction is really a key enabler in drug discovery. It gives us, on one hand, better understanding of biology. It allows us to begin to answer questions like, how do proteins interact with the cellular world around them? These proteins don't act in isolation. They form these interfaces with other proteins, creating these, DNA, RNA, small molecules, creating these amazing molecular machines. That is what, functions. And it really allows us to ask, what are these key interfaces? Then our drug designers can go in and think about how to create molecules that maybe disrupt that interface or maybe even strengthen that interface, depending on what functional output you want. And then on the other side, structure prediction allows us to do what I call rational structure-based drug design. So answer questions of things like, how will an introduced ligand interact with our target interface? How will changes in that design change the way this ligand physically interacts? So you can see this really nice example of, you start out with a small starting ligand, and you might want to grow this ligand into this pocket. And if you had a notion of structure for each change, you can imagine that your chemist is able to do this in a much more rational manner. And so our latest model, AlphaFold 3, is really, the next step in structure prediction for us. This is joint work from some amazing colleagues across Isomorphic Labs and Google DeepMind. We published this a couple of weeks ago in Nature. It's a single model that can predict the structure of proteins in complex with other proteins, but also DNA, RNA, small molecules, also things like ions and post-translational modifications. You can see some of the amazing structures that it produces in this video. And really the core of the model, it takes some of the amazing parts of AlphaFold 2 and takes these ideas a bunch further. Actually, in my mind, it simplifies a lot of AlphaFold 2 as well. It's now a diffusion model, for example. And this model sets a new state of the art for protein-protein, protein-ligand, protein-nucleic acid structure prediction. And crucially for us, it enables new approaches to do in silico drug design. So I'm going to talk about this model and some of the applications of it in the rest of the time. All right. So first, I'm going to do a little overview of the model architecture. This is the high-level neural network architecture of the model. There's a lot of boxes here and a lot of complexity underneath this, and we're trying to dig into a little bit of this, but I'd say it's probably quite dense. I can answer a lot of questions maybe after we've wrapped up the conference and later on, but there's lots of cool stuff in here. High level, this is what the model looks like. AlphaFold 3, at the core is a conditional generative model. The conditioning is provided by your inputs, which is the sequences of protein, the amino acid sequences for the different proteins or nucleic acids that you want to fold together with the SMARS strings of ligand molecules. These inputs are processed after featurization by a pretty chunky trunk, which probably forms the core of the parameters of this network. And this provides the conditioning to a diffusion model. And the diffusion model generates the output, which is a sampled structure of all atom coordinates, of all the 3D coordinates of every atom in the system that's specified as in the inputs. And you can see in the right, this animation shows, actually, this really is a generative model. It's sampling different conformational states of the system. How meaningful those conformational states are, I think, remains a big question mark, but it's cool to see this flexibility at least. In addition, there's a confidence head of this model, which takes the conditioning as well as the sampled structure, a particular sampled structure here, and produces a whole range of confidence values, which can be associated with particular residues, particular atoms, and the pairwise interactions between these atoms and residues. So in comparison with AlphaFold 2, this is the network architecture of AlphaFold 2, where you have your input sequence and MSA and templates. Then you have this evoformer and structure module with the dreaded iterative point attention. When we think with respect to AlphaFold 2, this basically replaces evoformer with pairformer and replaces this structure module with actually what I think is a much simpler diffusion head on top. So the first question that we had to ask when thinking about, okay, how do we put these different biomolecular types together in a single model is what is that computing unit? How do we tokenize, proteins, nucleic acids, small molecules, ions all together? This turns out to be fairly simple. A token is pretty much just a residue for standard things like standard protein, amino acids, nucleic acid residues. And then for things like ligands and ions, it's a single token per atom for a small molecule. And so we just concatenate, tokens from amino acids with tokens from atoms from small molecules if you've got a system of, for example, a protein and a small molecule. And then on the output space, we think about coordinates of these things. These coordinates are also ordered by token. You just have a fixed size array of number of tokens by number of atoms up to a maximum number of atoms where you just mask out the atoms that aren't used for that particular token. So if you've got something like an amino acid or a nucleic acid, you might have multiple atoms which have an X coordinate associated with that token. But if you have something like ATP, which is a small molecule, you'll have multiple entries for each atom, with only one X coordinates per token here. The rest is masked out. So in that way, we have sort of fixed sized objects that we can do, lovely compilable deep learning on. So the first step of the network is to do featurization of the inputs. These input sequences and ligands go through, something quite similar to AlphaFold 2. You have the genetic search and template search for proteins and nucleic acids. For small molecules, these are represented by first generating conformers with RD-Kit, and then embedding these things with the ligand embedder, which is this atom transformer. There's also a template module and an MSA module for both of those respectively. I think the MSA is much simplified compared to AlphaFold 2. A lot less processing there. So more of the processing of the evolutionary information is pushed into the shared trunk of AlphaFold 3. So that was one of the design decisions there. Then looking at that trunk, this trunk takes in a single representation, which is number of tokens by channels, as well as a pair representation, which is number of tokens by number of tokens by channels. The pair representation allows the network to represent the interactions perhaps between some of these tokens. And this pair former, on the single representation is essentially a transformer with an attention bias, which comes from the pair representation. And for the pair representation, the processing is a sequence of triangular attention updates and a transition. And you can think of this as like a simplified version of the Evo former from AlphaFold 2. And then finally in the network, you have the diffusion module. This module is doing Gaussian diffusion on continuous 3D atom coordinates. This builds on the really amazing work of Karas et al. From NVIDIA, that framework, but then applied to coordinates. So this paper elucidating the design space of diffusion models applied to image generation in pixel space. Actually, a lot of those techniques can be transferred into 3D atom coordinate space. There are some really nice tricks, things like skip connections between inputs and outputs, noise-dependent scaling of your outputs of your network, and initialization of your weights so that you get unit loss across all noise levels at the beginning of training. The conditioning to this diffusion model comes from the single embeddings that have been processed by the trunk. The pair embeddings that have been processed by the trunk, they get projected to these pair bias terms for the transformers within this diffusion model. And then the diffusion model itself takes in, has a series of attention layers. Some are local atom level attention blocks. So the model can attend to all the local atoms, I think within 128 atoms there. There's no split by chain here. So, you can actually attend, if you're in the local sequence vicinity between a small molecule and a protein, you'll be attending across atoms across these two chains. And then there's a whole, 24 blocks of global attention on the token level, followed by, again, going back to atom level attention. So it's almost a little bit like a hierarchical attention network here. So you have the noisy atom coordinates as input, the conditioning from your trunk, and then the denoise atom coordinates as the output. There's actually no equivariance built in to this model, which disappointed some people, but I would say there's a lot of equivariance actually built into the training process, which is, through data augmentation here. And then in terms of the losses in this space, for diffusion, you have the mean squared error on atom coordinates, as you would expect. Here you align the ground truth structure with your predicted structure. There's actually an extra loss that gets added in during fine-tuning to add extra weight to bonded ligands. And there's also a smooth LDDT loss. LDDT is one of the metrics that we're trying to optimize here for structure generation. And we create a smooth version which can be backpropped through. These are all added up and then scaled by your noise levels. So now I'll dig into the training details of this. So here's a little schematic of the full training loop of the model. Again, you have the network trunk here, which is takes the input and creates these conditioning vectors. You've got your diffusion module here. During training, for each input, we actually create a little mini batch of diffusion samples. So these are 48 different samples at completely different noise levels, all using the same conditioning and all trying to be denoised at the same time. There's the loss I talked about. This loss comes from the ground truth. Here, the ground truth, because we've got this bimolecular complex, which can have multiple chains, multiple copies of the same protein in this complex, you could have, you can also have these carbon rings where how you label the individual carbons of this carbon ring, you can start from any single point on that ring. There's quite a few symmetries and permutations that need to be resolved. So you sort of do that little search before you apply your loss function at the end, so you get the minimum loss out. There's also the confidence module, which is trained. This confidence module has to take in a structure. So at training time, there's a little mini rollout that happens, just 20 iterations of diffusion, 20 diffusion iterations that happens to create a rough structure that's fed into the confidence module, where we can apply our losses to predict things like LDDT. So there's a predicted LDDT looking at local structure at the atom level. There's sort of a matrix predicted distance error, which is the predicted distance between tokens I and j, and the error in that. And then the predicted aligned error, which is after you've aligned on a particular token, the error of the distance between two tokens. This is what training looks like, split by a whole bunch of different metrics that we measure during training. So you can think of these as different interfaces that we're training for. So you have protein-ligand interface, protein-protein interface, but also internal LDDTs for particular biomolecular types. So intraprotein, that just means how good are you at folding just a particular protein with respect to itself. Protein-ligand would be how good are you at folding a protein with respect to a small molecule ligand. You can see most of the action happens in training very early on, but there's a long tail of performance improvements, including these multiple fine-tuning stages. The initial training takes about 10 days, and the fine-tuning stages add things like increasing the crop size from 384 tokens to 640, then 768, adding this polymer ligand bond loss that I mentioned in the diffusion section. It also adds in this transcription factor distillation set, which allows the model to, forces the model to learn to recognize, where transcription factors will bind to particular parts of DNA versus not. Adds in things like more chains and also adds in more confidence measures as we go through these fine-tuning stages. And you can see the impact of some of these fine-tuning stages, I think probably most pronounced here on the bottom right, which is the, protein-protein interface LDDT. You can see that actually this is steadily increasing, and even in these fine-tuning stages, we're getting a bump in performance here. This is probably due to increasing the crop size in these stages, where you're seeing much more of the context and much more of the interfaces in these later stages. So now we train the model, we'll have a look at some of the results. These are on our test sets and some of the benchmarks that are out there in the world. I think across the board, the model achieves a new level of performance for all of these protein molecule interfaces. starting with protein ligand interface, we see large improvements in protein ligand structure accuracy. I think there's a big jump here compared to some of the docking-based methods. And I think critical to this method is it doesn't actually require any ground truth protein templates to dock against, so you don't need a ground truth structure here. The only input is the amino acid sequence. There's also the ability to leak some of the structure or leak some of the information into this model. And if you, for example, leak the pocket information or some of the pocket information into this model, you can get a pocket condition structure prediction, which is, again, has a bump in accuracy, but not too much, actually. Then looking at structures involving DNA and RNA, we see large improvements in protein nucleic acid interface prediction. For protein RNA and protein DNA prediction, we see actually doubling in accuracy compared to the other existing methods. And even in just sort of pure nucleic acid only structure prediction, we see quite strong performance here. This model is also able to handle covalent modifications. So this can crop in with things like post-translational modifications. There's a really interesting example here where you can see this is actually a complex between this protein over here and a peptide, which is this sort of wiggly worm-like thing at the top. This peptide actually has a serine which has been modified to this phosphoserine. And if you represent this peptide in AlphaFold 3 with the standard amino acid of serine, the model will actually place the peptide down here in the wrong position with respect to the ground truth. If you represent this properly as a covalent modification, then the model is able to pick up actually where this is meant to be and places this very, very good with respect to the ground truth. And you can see some of the side chain detail here, which is implicated in the binding mode of this peptide. And then finally, on the protein-protein class, this model is actually even improved slightly upon AlphaFold 2 in terms of just actually proteins by themselves. There's about a 10% bump in accuracy with proteins interacting with other proteins, so the protein-protein interface in general. I think what's really interesting, though, is that if you look at some subclasses of interfaces for protein-protein interactions, there have been big increases in accuracy. And one very notable one for therapeutic design is in antibody-protein interfaces, where there's been a big increase in accuracy with respect to AlphaFold 2.3 multimer. Also really interesting from my point of view is that for this model, you can run, because it's a stochastic model, it's a generative model, you can run a number of seeds, and with the confidence heads, you can pick out of those seeds the structure which has the most confidence. With antibody protein interfaces, you can see that this actually just keeps increasing in a log linear fashion with the number of seeds, which is quite cool. And then finally, for confidence, we see that this model produces a suite of confidence metrics, which actually quite accurately tracks the errors of the predictions that it makes. So here's an example with of protein-protein interfaces. On the y-axis is the .q score, which the higher the better. This is almost a measure of ground truth accuracy here. And on the x-axis is the predicted distance error that comes out of the model for this interface type. And you can see that as the predicted error increases, that .q score decreases. So the model has some self-awareness of where it gets things wrong. So, okay, this is all well and good, but so what? At the end of the day, for application to drug discovery and drug design, we can create these amazing test sets. We do a lot of filtering on these test sets to make sure that there's low homology to train sets, that we have lots of examples in our test set with completely novel interfaces. We create these general average performance metrics, and they look good. But at the end of the day, we need to observe a tangible translation into novel real-world examples that we work with into the lab. And the cool thing is we're actually able to see that generalization to completely novel cases. We're actually using this model day to day for real drug design on some of our partnerships with people like Novartis and Eli Lilly. So it's really exciting to see. What I will do is show some examples, some particular examples. These are case studies from literature which shows some of the novel generalization that we can get out of this model. Of course, these are cherry picked, but they show some of the novelty that's available. So in this example, here we see the model. This is an example which has a completely novel fold. This is a, an interface in a fold that isn't seen anywhere during training between two proteins with a small molecule in between, and the model is able to correctly identify that. Really interestingly, in this example, again, this is a complex of multiple proteins with a small molecule here. This is an allosteric binding site. So this is where this small molecule binds to this protein, not in the standard pocket that people know about, into a pocket that no one ever knew about before this paper. So the fact that the model, AlphaFold 3 is able to pick up on this binding mode, gives us confidence that we're able to use this model to design molecules for allosteric sites, which is of great interest for drug design. Here's another case study for another really challenging example. This is for a target called KRAS, which is considered an undruggable target in the field. In August last year, there was this amazing publication that came out which characterized a new potential compound which modulates this, and with that they published a structure. This is actually a molecular glue, so it's a small molecule that comes in, binds to KRAS as well as binding to this other protein CYPA, brings these things in complex. This is a completely novel ternary complex. This has not been seen before during training, and actually even more, this compound 1 has a covalent attachment which is not specified as the input. So the model is able to work out, actually, this model probably covalently bonds to the protein here. So the way that this is predicted is by taking the inputs, that would be the amino acid sequence of KRAS together with the amino acid sequence of CYPA, then put the SMILE string of compound 1, which you can see over here from the publication, together with the SMILE string of GNP, which is a cofactor that they crystallize this with. Now, again, because this model is stochastic, and this is a really hard example, we run this a lot of times, maybe 250 times, and we get a whole distribution of poses and structures out of this. Here in this graph in the bottom right, you can see every dot here is a different structure predicted by AlphaFold 3. The x-axis is the RMSD, the protein ligand pocket RMSD, with respect to the ground truth published structure. And you can see that there's quite a spread in RMSD here. So a lot of the predictions the model actually gets wrong, right? But the model also has a confidence head, which scores for each of those predictions, how confident am I in this particular interface? And you can see if we select the top 3 confidence head predicted poses, we get the pose which is published here. So, this gives us a lot of confidence in being able to use this for really challenging cases. And finally, I want to show this example. This is a target, again, a study that was published, a cancer target. Here, the authors are doing drug design step by step. So first, they created this ligand 1, which we folded, and we show really good quality with respect to the ground truth. Then they keep elucidating the design of this, so adding on atoms to make this a stronger and stronger binder in the publication. And you can see for this next step, the model is again able to predict the structure really accurately with respect to the experimentally crystallized structure from the authors. And then there's another iteration of design that happens, and again, the model is able to correctly pick up on some of the subtle changes here as we go through this design process. So again, this is a little bit emblematic of how we're using this model. We can use this iteratively for structure-based drug design. Now, of course, this is not a perfect model. This is a machine learning model. It gets things wrong all the time. There's a lot of limitations. I think some of the key ones from my point of view, first is that, I talked about this being a generative model. It can sample different conformations. That's all well and good, but actually the coverage of conformations can be limited. It depends on different situations. particularly if you've got examples when the training set, you'll see only one conformational state of a particular protein or a particular complex. The model is a statistical model at the end of the day, so we'll be biased towards that. So here's an example where the model is biased to the hollow state, the state which is bound to a small molecule, even though it's being predicted in the apo state. Another one of the limitations is, of course, this is a generative model. It's going to hallucinate things. And annoyingly, with AlphaFold 3, when it hallucinates things, it still tries to make nice secondary structure or plausible structures. So here you can see the AlphaFold 3 prediction for this protein. There's a lot of disordered regions of this protein, but AlphaFold 3 seems to be predicting some sort of structure here. It has labeled this as orange, which is low confidence, but compare this to AlphaFold 2, where it's quite clear that this is a disordered structure. And this is one of the limitations. Now we're trying to mitigate that by actually doing distillation of the AlphaFold 2 disordered regions into AlphaFold 3, which does mitigate this somewhat, but not perfectly. And finally, as we scale up the input to large structures, we can see that we actually get clashing of some of the molecular elements of these large structures. So for example, in this example, we have this self-overlapping of DNA, and also overlapping of two different protein chains, which is of course nonsensical. So where next for us? I started by saying structure is really important and critical for drug design. I say that actually structure is not the goal for us, right? It's actually just a proxy for function for us, right? Structure helps chemists rationalize drug designs through the interactions that are being made with these proteins and these systems. But really, at the end of the day, what we want to do when we're designing a drug is actually modulate the function of these proteins in a cell, right? So for me, the big question is, well, how do we move towards models that actually predict function and predict modulation of function rather than just static structure? So, I think it's an open question for us, like, what are other good proxies? Could it be things like modeling the change in structural state with respect to designs rather than the absolute state? Is it things like modeling dynamics of the system rather than the crystallized, idealized forms of these systems? Also understanding the change in the wider cellular interactions that occur when a particular biomolecular system is changed in a particular way. Ultimately, at the end of the day, we want this hypothesis generation tool. It's all about change. If we make a change to the system, what effect does this have? Not just on the structure, but actually on the function. And ultimately, how do we move towards seeing this on a cellular level? I think it was really amazing to see the talk on all the imaging work. And as we think about introducing small molecules into that imaging pipeline, we're going to start seeing that change on the imaging level. Can we start connecting these dots a little bit? I think there's a real opportunity here. I'm also highlighting this work, amazing piece of work from Google DeepMind on alpha missense, which again starts to look at this using structure, and you can see how the change in particular residues affects not only the structure but also the pathogenicity of that mutation. Great. So just to wrap up, I want to emphasize that there really is an amazing potential here to apply machine learning to have tangible impact today on drug design. We're seeing that day to day. Understanding biomolecular structure is a key tool to help us do this. It helps us learn how our molecular machines may work, and helps us rationally develop modulators, i.e. drugs, to change their behavior. We developed AlphaFold 3 to expand the breadth and the accuracy of biomolecular structures that we're able to predict. This is primarily by being able to tokenize all of these different biomolecules into a common format, diffusion modeling the 3D coordinates for generation, and then training on all of PDB. We're seeing great performance on general benchmarks. Also, critically, we're seeing really interesting performance on therapeutically relevant novel cases. And this allows us to incorporate AlphaFold 3 and models like this for day-to-day drug design. And I just want to finish by saying it's really exciting to see how we, machine learning researchers, actually have the potential to move the needle forwards in our ability to understand and modulate biology. So I'm just fascinated to see how this field progresses, over the next 5 years. I think it's going to be like night and day as we make these sort of advancements together. Yeah, thank you.
Thanks a lot for the amazing talk. It was really interesting to learn about AlphaFold 3 and especially something that was released so recently. I think it's fair to say that AlphaFold 2 was, I don't know, like was a breakthrough that boosted a lot the field of machine learning for drug discovery. And it's great to see that we can still improve on that. We can still progress on that. But that there are other steps that are required to make everything work. So it was a great presentation, and if there are people who want to ask questions,
yeah, a lot of enthusiasm from our side.
So, you know, if you think before AlphaFold, what people were doing to combine evolutionary and physics was like a two-step process. And from the MSA, they estimate coevolution to predict contacts, and the contacts go into a physical optimization. You know, AlphaFold 2 almost looked a little like that. AlphaFold 3, as you pointed out, the MSA embedding is tiny. So the evolutionary information is just going into this pairformer, and we don't know really if it's using evolutionary information or if it's doing physics inside the pairformer. Is there any way to dissect it and understand how it actually works?
Yeah, I think understanding how it actually works would be, you know, really interesting. I think it's fair to say that there's a lot left unexplored. The MSA is very, very useful here. So, you know, if you remove it, you decrease performance. It's clearly using that evolutionary information. I also saw some great analysis by Sergio on like, you know, how this new, you know, MSA module actually like approximates some of the more traditional analysis you might do on these MSA matrices.
Interesting. Who was that?
I can post some of this later, perhaps on the portal. But yeah, I think, you know, exactly what is learned in this network, like how much of it is physics, how much of it is evolutionary information, is very hard to disentangle this. You know, we do see great generalization to completely novel cases, which would, you know, obviously indicate that there's some extrapolation, some notion of physics there. But at the same time, we see a lot of biases with respect to training data. So, you know, it's still a statistical machine. Yeah.
Thank you so much for the great talk. I was wondering how much data augmentation went in removing equivariance from the whole training process.
Yeah, so you know, when I say data augmentation, that basically means that when you're doing diffusion, you sort of randomly rotate and translate your complex. That's it. It's not. It's you know, it's nothing too crazy. You just do that for every single sample. As I mentioned, in every forward-backwards pass, you actually have this mini batch of diffusion model samples where you have an extra 48 different, you know, random rotations and translations, all at different noise levels, which get processed simultaneously, and the loss is applied and backdropped into the conditioning module, into the trunk. So it's just that random rotation and translation that gives it the equivariance in that SE3 sense. Yeah.
And do you need to do that at inference time?
At inference time, you would also randomly rotate. That's part of what the seed will control.
Okay, and what was your thought process about removing equivariance from it, given all the previous work about like how equivariance make training more data efficient and also inference more data efficient and all that?
Yeah, I think this is purely empirically driven, right? So what works best was this model, which without equivariance. I'd say, you know, the recurrent message that we need to take to heart with deep learning again and again is, you know, when, you know, neural network modules and layers which allow you to scale the depth and the size and the number of parameters when you're in that, you know, information rich and information dense regime that I think we are in structure modeling. Those are the things that end up winning out. Now I'm sure there's an equivariant version that might do a little bit better as well. But yeah.
Thank you so much.
Thank you for this clear explanation of the AlphaFold 3. I'm curious about the evaluation. So I like the use cases you mentioned. And I think for any machine learning model, it's important to actually evaluate the real scenarios. So do those use cases include any wet lab experiments?
So the
Yes, the use case you mentioned, or maybe the other ones that are based on AlphaFold 3.
Yeah, so here the wet lab experiment is actually, crystallography, X-ray crystallography, which can take, a long time to get that full structure. Now, maybe you can you can maybe verify a structure in a cheaper manner with, there's a bunch of techniques to actually verify, whether you have an interface correctly predicted or not, but not get the full structure. So you could think about maybe doing that to experimentally verify in a cheaper way. But to get the full structure, I mean, it's still something like X-ray crystallography or cryo-EM, which can take a long time. Yeah.
Oh, well, I was thinking about a ligand protein interactions and actually maybe measuring some binding affinity. These kind of things have they been done?
Yeah, so you can think about if you're using this model for drug design, you might make, you have this predicted structure, of something that binds, you make some changes as a medicinal chemist that you think, will create new interactions. You send that new molecule off to the lab, you get it back, and the binding affinity is increased. Okay, maybe that confirms your hypothesis, you're making these interactions, but it doesn't, you can't conclude that, I guess. Yeah.
I see. Well, thank you. That's clear.
Thanks for the talk. Really, really exciting stuff. I was mostly wondering, I'm under the impression that you did a very, I guess, extensive analysis of protein ligand binding with hostbusters. And I was wondering if you could speak a little bit about the failure modes. So if most of among the examples that failed, was it more of a failure mode that had to do with the ligand conformer itself, so in space, or maybe like invalid bond lengths or angles, or if it was more to do with its pose within the binding site of the protein?
Yeah, so I think there's a few different failure modes that we see. A little bit of failure mode on stereochemistry, which is always this recurrent thing. A little bit of failure mode on like some of the bond angles. But then, really when we look at the performance of this model, ligand protein, it's really the novel category where you have a complete, a novel ligand with a novel protein. There, the performance is lower than if you had any known element of this. So I think it's, there's always a decrease in performance with respect to novelty in general.
Okay, so is it? I don't remember exactly if you had that, if you mentioned that in the paper or not. Is there, so how significant is the drop off when you have, say, a novel ligand and a novel protein compared to if one or both have been seen before?
I can't remember the exact numbers. It's, something on the order of like 10% or something like that. Yeah.
Okay, thank you.
Hi, thanks for the talk. You mentioned that AF3 is way more stochastic than AlphaFold 2. And I noticed that in the protein-protein benchmark that you were reporting, you ran inference 1,000 times and kind of selected the best pose from that. And similarly, in the KRAS example, I think it was 250.
Yeah.
Right now, the daily cap for requests is 20 for public access. And I was wondering if there are any plans to kind of increase that cap or just generally make the model more accessible.
Yes. So, with respect to the cap, I don't know how this will change over time. On the server, we'll be releasing, the model weights and inference codes, in due course, which means that, people can just run this as much as they want for academic use. Yeah.
So, thanks.
Hi, thanks so much for the talk. I had two quick questions. One, I was curious if you saw the blog post recently by Inductive, the strong baseline for AlphaFold 3, where they used like multiple conformers in Vena and then Vena re-ranking and got similar performance but with rigid docking. I was just curious what your thoughts were on that as a baseline.
Yeah, I think there's, we've done tons of analysis internally as well on docking methods using, lots and lots of compute on docking and ensembles of different methods. I'm sure there's going to be many ways that we can start to like reach this performance with other methods. That's kind of the exciting bit about this, right? And I think there'll always be a space for docking because of maybe some of the, at least for now, like how computationally fast it can be. But if you don't have that decent template to dock onto, or you don't know, with docking, you need to know the pocket information because you need to draw your box that you're sampling from. If you don't have that information, what do you do? Again, there's probably heuristics that you can do, but this offers another framework that you can try out.
Thank you. And then the second question was, I guess, how do you see this being used in small molecule drug discovery? As far as, is it more so this like iterative approach where chemists are testing theories of like, oh, if we have this functional group, how does it change the conformation, and iterating on it like that? Or do you see it also being used in large scale screens? Or do you think that the current compute requirements are a limiting factor for that?
I think there's many ways this could be used, and I don't think I think it will take a long time to sort of explore that long tail of creative ideas of how we can use this. I think we saw with AlphaFold 2, there was a lot of what I call dark knowledge in this model, right? Ways that you could start extracting new types of function, maybe with a bit of extra, playing with your conditioning, maybe with a bit of fine tuning over here and there. I'm sure there's a lot that can be done here. I think yes, we can use this for like iterative drug design, like I showed. Another interesting case is you found a binder, or there's a literature reported binder, but no one knows how it functions. Then you can, maybe start to get a picture of what pocket it's targeting and what are the key interactions or the key states of this system. And given enough compute, yes, you can run this for large scale screens as well.
Thanks so much.
Yeah, so this will be the last question before we close the conference.
Hey, thank you for a great talk. Any comments on how these opportunities and potentials generate to membrane proteins? Because membrane proteins are a lot harder to get structures, and also resolution tend to be lower.
Yeah. Membrane proteins definitely fall in that sort of harder category of things to model, where the general performance will be lower of this. But there are still quite good success rates here, modeling things like GPCRs. I saw some really cool community experiments where people put in lipid-like molecules and actually started to see that how these proteins sit with respect to the membrane. So yeah, this still can be used. Maybe the performance is lower than the top line average performance, but not disastrous.
Okay, thanks a lot, everyone, and another round of applause for. Right, so please stay seated a bit for the closing remarks. First, I want to say a few words about like where you are right now in and why you're all present here. Again, like we mentioned the sponsor during the opening, but it's something that's more important than the sponsors is the machine learning community that is here in Montreal, and MILA, which is like the Quebec Institute of Machine Learning, which built this amazing community that allows like to constantly push the boundaries of what's possible with machine learning. So, and they range over like tons of different applications. They have hundreds, or if not 1,000 people affiliated to MILA here, both from industry and from academia, both at the PhD level, but also a lot of postdoc and professors as well. And MILA has allowed us like to do this conference today and helped us organize this conference today so that we can bring a lot more of the talent that is out there here and like discuss about the recent advances, especially in fields that are really important, like healthcare, about like curing diseases. Everyone here could be doing like some TikTok filters, but we're here to like cure diseases, and not just disease, but also design new materials for the future. There's a lot of opportunities like in molecular discovery, and it's great to be able to like unite all of us today. And also part of the community is not only about MILA, but also a virtual community that we have been building over the past year to unite many people that work at the intersection of molecules and machine learning. And here we can see this the portal. If you can join the portal, we have different reading groups. We have blog posts by various authors, internationally recognized authors, a lot of discussion, and a lot more than 1,000 people affiliated to that community that actively discuss the recent papers, the recent works. And all of these reading groups also have YouTube channels associated to them, where you can on a weekly basis learn about the recent advances in the field. So yeah, this is the YouTube page where you can see like every week there's roughly 2 or 3 videos that get released that discuss in-depth topics and in-depth papers. So yeah, subscribe here for the newsletter to stay in touch with all the recent things that get posted. And now, as the last remark before everyone leaves, I'd like to have everyone come here in the front so we can take a big group picture and showcase showcase that we had fun here at the conference. So I want like happy faces, hands in the air. We can stay in front since we'll try to put all the speakers in the front.
Yeah, it's going to be a challenge. I think we have to. There is good in new compress. Maybe I think it's on. Good for the fair. They do it. Sorry, guys.
Come on, guys, give me a nice smile. Come on.
Hi. Just returned in time. I just returned in time. When are you flying tomorrow? Or tomorrow?
Yeah, tomorrow. I leave at 4. I think I get in like an hour before you, but I don't know how hard it is to get a taxi, so I might just wait until you plane land.
My finance.
Yeah, you get in like an hour after.
Okay.
I'll message you.
Sure. If you want to wait for now, yeah. Yeah. All right. Yeah.
We'll see what the group looks like at 8 o'clock tonight.
Yeah. Are you flying there, right? Yeah, nice.
That's all I got. The model's still training, so.
Yeah.
Still waiting to get back to Paris. It's very optimistic.
I want to see like should I give you some stuff? Like I was thinking, I was wondering myself if I should do some coding with the stuff. Yeah.